St. Joseph's College Autonomous would like to welcome all of you to the two-day Science Academies lecture workshop on recent trends in microbiology and biotechnology, organized in association with the Department of Biotechnology, St. Joseph's College Autonomous, Bangalore. Invoking God's blessings on each of us and hoping that this workshop is informative and fruitful, I would like to begin the inaugural session. I request Dr. Madhappa MB, HOD, Department of Biotechnology, St. Joseph's College Autonomous, to deliver the welcome address.
Thank you, Andrea. Good morning to one and all. Thank you for associating with us for the Science Academic Lecture Workshop, Recent Trends in Microbiology and Biotechnology. As we know that this is a collaborative effort from the Indian Academy of Science, Bangalore, the Indian National Science Academy, New Delhi, and the National Academy of Sciences, Allahabad. The Department of Biotechnology, St. Joseph's College, is very happy to be associated in organizing our second Science Lecture Workshop series. Well, it's a cold day, and if you all were here, I would have welcomed you all with a hot cup of coffee and maybe idlis from our canteen. But I have to just give a warm welcome to our convener, Dr. Dipshikha Chakrabarti. Ma'am, good morning and welcome. Ma'am is a professor from Center for Infectious Diseases Research, IISc Bangalore. Ma'am, thank you for your efforts in making this happen. On behalf of the management, the principal, the staff, and the students of St. Joseph's College, a very warm virtual welcome for you, ma'am. And I hope we meet you soon at our campus. I also welcome our coordinator, Dr. Grace Prabhakar. I extend a warm welcome to Dr. Bhagyaraj, Dr. Harinikumar, Dr. Chandrayappa, who's going to be also our keynote speakers in the days to come. And dear participants, past few months has been, we can say, a domesticated virtual overdose for us. From small, my grandmother used to tell me, too much is too bad, but the next 2 days is going to be too much and it's going to be too good. So I welcome you all for the Science Online Lecture Workshop series. And I hope we should be having a good science field and a lot of learning to be there for the next 2 days. Thank you again. And I hope to have a fruitful and a successful event in these 2 days. Keep your ears open, and thank you for associating with the Department of Biotechnology, St. Joseph's College. I wish all the success to the coordinator, Dr. Grace Prabhakar, for the two-day event, and also again welcome you all on behalf of the management and principal. Over to you, Andrea.
Thank you, sir. I now request Dr. Dipshikha Chakrabarti, convener of this workshop and professor at the Department of Microbiology and Cell Biology, IISc Bangalore, to present the opening remarks. Over to you, ma'am.
Oh, thank you, Andrea. Thank you, Dr. Grace and Dr. Madhappa, for such a very nice and warm welcome. Yes, indeed, Dr. Madhappa, I do miss the coffee from St. Joseph's College. I had been there and I still remember and cherish those wonderful moments that I had with all of you and really your all young colleagues. So, of course, this is the first online academic lecture series that we are organizing. And I'm very happy to be associated with St. Joseph's College and the first online lecture series under these prevailing situations. And we would like to make it really a very different experience. I know very well that the young colleagues with whom we always love to interact, they are on the other side behind the screen, and that connectivity may be lost somewhere, but we will try to see that we make that connectivity very strong, and each lecture will be making them connected to more and more and maybe learn about more and more. So we will try our level best in that. So before we begin, let us pay our gratitude to all the three academies. And I would like to tell you a bit about all the three academies so that you get all the advantages of the work that is done by the academy to promote the education. So I will share my screen now. Can you give me the control over sharing screen? Okay, thanks. I got it. Yeah, thank you. Thank you, Grace.
Most welcome, ma'am.
Trust that you can see my slide.
Yes, ma'am.
Okay, thank you. So let us talk about these 3 academies in very brief detail so that all of you and my young colleagues, they get to know about all the programs that are being conducted by the academy and how they can benefit. And you as a very precious teachers of the college, how you all can get benefited by these academies. Now, as Dr. Madappa mentioned, the 3 academy, Indian Academy of Sciences, Bangalore, National Academy of Sciences, Allahabad, India, and the Indian National Science Academy, Delhi. These 3 academies play a very pivotal and a crucial role in developing the education panel, which spans across not only inducting fellows to these academies, but also to make sure that all the budding scientists and the young minds which have the inquisitiveness to learn, they become very, very much happy to see that there is a way for them to get inducted. Now, a brief about the academy. The Indian Academy of Sciences was founded by Sir C.V. Raman in 1934, a Nobel laureate, and its objective include promoting the progress and upholding the cause of science, both in pure and applied branches. So this particular academy deals with also the translational science and of course the pure science. And in 1994, the Academy brought out a document on university science education, and it contained several recommendations to improve the state of science in the country. So having said that, the Indian Academy of Sciences have played a very important role in shaping up the Indian science, especially in Bangalore and across India. Now, the second academy is the National Institute of Sciences, which was known as it is the Indian National Science Academy, which is headquartered in Delhi now. But if you really look at the history, it's very interesting. It was known as the National Institute of Sciences of India and was instituted in 1935, 7th January 1935 in Calcutta, and where it functioned as its headquarters at Asiatic Society for Bengal till 1951, and thereafter it was shifted to Delhi. Now, Indian National Science Academy was established in 1935 with an object of promoting science in India and harnessing the scientific knowledge for the cause of humanity and national welfare. There are 3 journals which are published by the INSA, Proceeding for the Indian National Science Academy. This is an amazing journal. I would request if all of you who are doing research here, you should try to put your findings in this particular journal. This is a very high-hold journal with a lot of prestige. The Indian Journal of Pure and Applied Mathematics and Indian Journal of History of Sciences. Now, the third academy that I would like to talk about is in fact one of the oldest academy, the National Academy of Sciences, India, Allahabad. Now, this National Academy of Sciences, the founder president is Professor Meghnad Shah, an amazing physicist. The Academy of Sciences of United Province of Agra and Oudh, that is how it was called. It was founded in 1930 with again an objective to provide a national forum for the publication of the research work carried out by Indian scientists and provide an opportunities to exchange views. Now, all these 3 academies had only one motto, that is a promotion of science. And you may notice that these 3 academies, they come together and fund, but specifically fund with the terms of money to get going with all the knowledge that we can share with the young budding scientists. Now, the Indian Academy of Sciences publish a journal called Resonance. Now, this journal, Journal for Science Education, is published monthly and was launched in 1996. And I would actually advise or maybe ask all the students, whether you are in the graduate or undergraduate or in school, you must go through this journal Resonance, because this journal is an amazing journal which gives you an articles for you to read and tinker your thoughts. You will have the articles in the mathematics, in the physics, in the biology. They are written in a wonderful story fashion, and it is an amazing journal to look into. So all my young colleagues, my young students, kindly read monthly these resonance journal. And in fact, we had many of the colleges, they have subscribed to this journal. And many of the colleges, we have made the subscriptions free for them. I mean, from our side, so that you can get an access to the journal. Indian Academy of Sciences host all these journals, right from the Journal of Genetics to the Proceedings of the Mathematical Sciences to the Current Science. And I'm sure that all of you know about this Current Science. Again, an amazing journal where each one of you who are doing your research, kindly try to see if you can put your research in these journals, which will indeed give you a platform to exchange views and ideas. Now, the most attractive and the hallmark program is the summer fellowships. Now, summer fellowships, which are hosted by the 3 academies, are awarded to very bright students and motivated teachers to work with the fellow of the 3 national academies on the research-oriented project. Now, all of motivated teachers of St. Joseph's College and many other colleges across Bangalore kindly fill this up and do not miss the deadline. Because if you miss the deadline, then we will lose the chance of getting you in the lab and allotting you labs. This is a highly prestigious, but at the same time highly challenging, because each day, each year, we see more and more applications. So it becomes very difficult. Bangalore is a wonderful place, but it also got a tag of having so many institutes around. So maybe you may notice that you may miss the chance because you're already in a place which is surrounded by institutes. And the summer academies and summer fellowships are in fact for the students and the teachers who are in the remote places where they do not get a chance to interact. In your case, you can always come over to IISc, meet me, interact with me, my colleagues. You know, there is absolutely no barrier. But for the teachers and the students who are really very far away, for them, it becomes a problem. So in fact, the summer fellowships are meant, but of course not restricted. So please do not miss your chance and kindly apply for these summer fellowships. Now, the refresher courses for the teachers. Now, this is an all India program to help the motivated teachers improve their background knowledge and teaching skills. It's a 2-week duration course, and approximately 30 teachers all over the country, they undergo a very rigorous course of lectures. I'm sure that maybe 1 or 2 of you have got a chance in the previous teacher refresher course for the teachers. If not, kindly keep applying, because definitely it's an equal distribution. Here, there is no discrimination of the areas, and you get an equal distribution of Bangalore, the Karnataka region, and all over the 22 states of India. So this is an amazing course for the teachers to get, abreast of all the recent knowledges. Now, the lecture workshops. The lecture workshops are again one of the most attractive feature of these 3 academies. The academy arranges 2 to 3 days lecture workshops in a very carefully chosen topics of any discipline, physics, mathematics, chemistry, life sciences, in universities and colleges. And these lecture workshops, in fact, gives a very strong bonding and connectivity between the young minds and the experts in the field, which they would have missed to interact with. And because the students are in very high number, the mass number, it becomes extremely important for us to organize these lecture workshops to bring them much closer to us. Now, the feedback sessions, which is also very encouraging, and it is very positive because your feedback gives us a thought, your feedback keeps us going, and your feedback is extremely important for us to make us perform better and also grow better. So me, on behalf of all the 3 academies, because fortunately I am a fellow of all the 3 academies, and on behalf of that, I pay my gratitude to the 3 academies, and I pay my gratitude to all of you who are arranging these lectures so that we keep going, and your feedback keeps us more and more better. So thank you all, and I stop here. Over to Grace.
Thank you so much, ma'am. Thank you.
Yeah, thank you so much, ma'am. I now request Dr. Grace Prabhakar, coordinator of this workshop and professor at the Department of Biotechnology, St. Joseph's College Autonomous, to give us an insight about the lecture series. Over to you, ma'am.
Good morning. St. Joseph's College Autonomous is managed by the Society of Jesus, or Jesuits, a Catholic religious order founded in 1540 by St. Ignatius of Loyola. The college was founded in 1882 by the fathers of the French Foreign Mission and bears a 139-year-old legacy of creating men and women for the society. The college is registered under the Bangalore Jesuit Educational Society. It is re-accredited with A++ grade and a 3.79 on 4 CGPA by the NAC. The college is recognized as a College of Excellence by the UGC. The education imparted by the college enables the students to strive for both academic and human excellence. Academic excellence is pursued by providing a learning environment that constantly challenges the students and supports the ethical pursuit of intellectual curiosity and ceaseless inquiry. Human excellence is promoted through courses and activities that help students achieve personal integrity. The college strives to achieve holistic development by encouraging the students to participate in activity and need-based learning. Established in 2002, the Department of Biotechnology has grown and contributed to the field of life science education. The department seeks to introduce students to the joys of learning science, to inspire students to inquire, imagine, and excel, and to equip them to integrate this learning into living. The department seeks to provide students with wholesome education in biotechnology, rooted in experiential and evidential learning. The current world scenario is gripped with the onslaught of the pandemic caused by COVID-19. There are graver situations of health, poverty, drought, malnutrition and undernutrition, energy management, amongst others. The current workshop focuses on how the science of microbiology and biotechnology can be used to address these burning issues. We also aim at creating a platform where students and faculty can interact with eminent scientists on these themes. Wishing all of you have a very fruitful and science-filled workshop these 2 days. Thank you very much.
Thank you, ma'am. I would now like to present the rules to be followed during this workshop. All participants are requested to kindly mute their audio and turn off their video when they join and throughout the sessions of the workshop. All participants should fill the feedback form, which will be sent after each session in order to receive the e-certificate. Relevant questions can be asked during the Q&A session at the end of each talk. And kindly avoid introducing yourselves on the chat box. Thank you. I now request Avantika Kamath to introduce the first speaker for the day. Over to you, Avantika.
I, Avantika Kamath, on behalf of the students and faculty of St. Joseph's College, would like to welcome the first speaker for the day, Dr. Dipshikha Chakravarti, to the lecture workshop. Dr. Chakravarti is currently a professor in the Department of Microbiology and Cell Biology at the Indian Institute of Science, Bangalore. She did her bachelor's and master's in microbiology in Nagpur and her PhD in biotechnology from Pune University. She was a fellow at the National Center for Cell Science, Pune, and a visiting scientist at Aichi Medical University, Japan. She was also the Humboldt Fellow at Institute of Clinical Microbiology, Immunology, and Hygiene at Friedrich Alexander University, Germany. She joined the Department of Microbiology and Cell Biology at the Indian Institute of Science, Bangalore, as an assistant professor in 2004, and is now a professor at the same. She has been awarded the Astra Chair Professorship in 2018, NASI Reliance Platinum Jubilee Award in 2015, the National Bioscience Award in 2010, and the DAE SRC Outstanding Investigator Award in 2015, to name a few. She has also attended various renowned national and international seminars. We are honored to have you, ma'am. Thank you.
Thank you, Avantika, for such an amazing introduction. Yeah, it is always nice to see that how our young colleagues are really so professional in making this conference happen. And it's all done by them. So it's a very proud moment to see that. I like the word that Grace mentioned. It's a holistic learning. Indeed, the the concept of holistic learning, which is introduced by the Jesuits and by the various other bodies of learning, it has a very deep meaning. So holistic learning is something very, very different from what we learn. But I always believed in holistic learning, and today is also the birthday of Swami Vivekananda. And Swami Vivekananda's teaching and believing in holistic learning actually really goes together. So today is a wonderful day that Swamiji's teachings, as well as the holistic learning and our workshop coincidentally falling on this date, is an amazing celebrations of many, many factors and many, many religions. So what I'm going to do is I'm going to take you through some of the fascinating and amazing story of bacteria, where we will realize that though they being so small and being invisible, how they in fact learn about us. Are we able to learn the same about them? That is a question. How they learn so much about us that they can bring us down, but have we done enough to learn about them, to really know them better? So let me share my screen and then we can begin. Regarding the questions, this is a workshop. It's a lecture workshop. So ideally, we should have had a very important discussion session where the interactive session where you can raise your hand and ask questions anytime that you want. So what we can do is you can share your questions in the chat, or you can stop me, show up, turn on your video and ask questions, whatever you may feel like. Do not hesitate to ask questions, because the very fact that these lecture workshops are arranged is only for asking questions. That is the most important thing. So I would request you to ask questions, because that is indeed very important. You can stop me, ask questions, or you can tell me that you can just stop me, or you can put it in the chat box. So now let us begin. Can you see my slides? Hello?
Not yet, ma'am. You've started
Not
screen sharing, but not yet.
Okay. Should come.
Screen.
Not yet?
Not yet, ma'am. Not yet.
1 minute. Sometime I have an issue with the PPT slide sharing where I cannot actually go to the full screen mode. That is the issue. Now let me check.
Not yet, ma'am.
Okay. How about now?
No, ma'am.
Your screen share is paused. I don't know why. Resume share. Okay. It is showing your screen share is paused.
Ma'am, I've given you the control.
Yeah. Now I wrote resume share. You can't see it yet?
No, ma'am.
Oh my God. Okay, let us try again. Okay, how about
Yes,
now?
ma'am. Yes, ma'am.
It's okay, great.
Yes.
So what I'm going to do is we will, now let me put it in full screen and just check that whether you are still able to see my full screen. You're able to see my full screen?
Yes, ma'am.
Okay, then we'll go like that. And in case in between you have any problem, my voice not clear or anything not clear, kindly stop me. Grace, you can do, even Andrea, Avantika, Dr. Madappa, whoever can do that. Yeah.
Sure.
Or you can call me in my mobile. Anything is okay. Yeah. And okay, so good morning, my dear young colleagues. A normal routine time, I would have been with you and presented this in front of you with a lot of very vibrant discussions over the coffee, over lunch, and over, small breaks. It is always very enjoyable, but now under the prevailing condition, let us enjoy this. All of you take your cup of coffee in front of the screen and sip and listen. Just lay back, lay down, and and just listen. And anything that you would like to stop me, ask me, kindly feel free to do that. Never hesitate to ask questions. So today I'm going to talk to you about a bacteria which lives in an intracellular compartment. The bacteria, and hence the title is a life in a vacuole. Now it looks like that today we are also kind of a life in a vacuole. Dr. Madappa mentioned about really this kind of a domesticated online fatigue that we are carrying. It's actually in a vacuole. We are in our own vacuole. I am not very sure when are we going to break this vacuole, but this vacuole was very important for us to sustain our life. And that was the need of the hour. So the life in a vacuole, sometimes it's very important, as it is important for certain bacteria. They just don't want to break the vacuole. Now, how is this life important and why do they vary so much among bacteria? That we are going to share. So in a nutshell, in my lab, we work on bacteria, different bacteria, vacuolar bacteria, cytosolic bacteria. But the major model system that we are working on is with Salmonella. Now, Salmonella is a pathogen, and the Salmonella is such a wonderful pathogen that it has not left any kingdom, including plant. So we try to understand its metabolism, its pathogenesis, and then try to understand that how we can design the therapeutics. The trans-kingdom invasion of pathogens, that means Salmonella in the plant, also is one very important branch that is emerging from our lab. All these three, it bridges the gap between the metabolism, the pathogenesis, the ecology, and the therapeutics. We will understand. So as I mentioned to you, that these are small bugs. They have understood us, such supreme creature with more than 30,000 genes. Are we able to understand these invisible creatures with 3,000 genes? Have we understood enough? That is the very interesting and major questions. And being a microbiologist, all of you, some of you are also a microbiology with the microbiology expertise, always ask this question, that are we good enough to have understood as they have understood us? So as a microbiologist, I am very proud to say that the bugs are somehow much superior to us. And if I put myself in their shoes, I can understand the swiftness and the passion with which they were to understand the host. But have we done that? Have we done that? Let us understand. So this is how the work goes in our lab. I will give you a brief description of how Salmonella gets inside. Each one of you know about the several route of infections. That means some bacteria prefer the route of aerosol mediated infection, that is through the air, through the droplets. Some bacteria prefers to go through the cracked skin, intravenous route of infection, through any wounds, through any openings. Some bacteria, they go through contaminated food and water. Now this is the oral route of infection. Now all the gastrointestinal diseases are taking the oral route of infection, that is through contaminated food and water. And certain airborne bacteria like Mycobacterium, they take the route of aerosol. Our own very famous SARS-CoV-2 or the COVID takes through the aerosol route of infection. There is no other route of infection which is yet identified for the COVID virus. So similarly, Salmonella gets inside through contaminated food and water. Now, how it gets inside, it's very simple. If we ingest the contaminated food and water, it passes through our food pipe, through the food tube, and then crosses the gastric acidity, and it has also the capacity to neutralize or to withstand this gastric acidity, pH 2.5 roughly, 2.5 to 2.6, and then it lands up in something known as a small intestine. Now, if you see here, the small intestine is a niche for the bacteria now to get settled. Small intestine is again one of the most important organ that shapes us. And why I say so? Because the small intestine is a hub for the gut flora, the various parts of the small intestine, the duodenum, the jejunum, the ileum, and the colon, and later the colon. They are the majority of the gut flora hub, and also they are the one which secretes a mucus. Now, this mucus lining, which lines the small intestine, protects our gut from any harmful pathogen. And on top of these mucus lies your gut flora. Now, the gut flora is very diverse. You have 230,000 different species of the flora, which is made up of a bacteria, certain viruses, certain bacteriophages, certain yeast strains, etc. So it is a very diverse flora. Now, you imagine the plight of a pathogen. And that is why I always say that they are far, far more intelligent and they can bring down our intelligence in no time. The plight of the pathogen is, it comes in through these orophycal route. It withstands the gastric acidity where everything can be killed in pH 2.5, but it withstands, and then it lands up in the small intestine. Now, the small intestine, they have this very thick mucus on the top. So here you see these enterocytes, these beautiful villi, and on top of that, you have those thick mucus. Now, the thick mucus is so thick, that is, the viscosity is so thick that in fact, the pathogen has to spend a lot of energy if it has to, degrade the mucus and then get in touch with these enterocytes here, and then take its route into the invasion process. Salmonella is far more intelligent. And why it is intelligent, I will tell you. It takes its entry through a certain cell type known as a M cells. Now, M here stands from the membranous cells, or it is M cell or M fold cell, because when you see a section of the intestinal cells or the enterocytes under electron microscopy, you see that it's around 0.1 to 0.2% of the entire enterocyte lining. They are lined by these so-called M cells. The ontogeny of the M cells is still actually not known. Way back in 1980s, 1970s, Maria Neutra's group, they worked a lot to understand how is M cells generated? What are the triggers for the M cell? Is one of these enterocytes getting converted to M cell? Or it is a certain very different cell type which comes in during infection process? And how bacteria or many viruses, they use the M cell as a portal of entry. So this is one of the most important portal of entry for the pathogens. And how it becomes very important? Because the M cells throw out their dendrites, so much so that they raise above this mucus layer. So now the pathogen has a very simple life. They don't have to fight with these very thick mucus and the gut flora. They simply slip through or slide through, and they land up in this cavity of these, what the M cells have created, and then they are secure. They undergo this process of transcytosis, where they are taken up by these M cells. You see here that the bacteria is getting taken up. So Salmonella is a gram-negative bacteria with a peritrichous flagella. Peritrichous means that they have flagella all over. So actually their motility or any motion is highly vigorous. That means they have so many flagella to make them swim across. But again, they are too smart to not tamper the mucous membrane, the laid mucus or the gut flora. They simply slip through this M cell. and then they undergo the process of intake or the uptake, receptor mediated uptake, where they land up in these so-called vacuoles. And from the apical site of these M cells, they are transcytosed to the basal site. Now you try to visualize or imagine this internal layer of the intestine, which is known as a lamina propria layer. Now the lamina propria layer actually is made up of a resident fibroblast and lot of cells like neutrophils, the T cells, the B cells, the dendritic cells, macrophages, everything constitute the lamina propria layer. There are many migrating cells that comes to these lamina propria layer, like gamma delta T cells and other, the neutrophils upon infection. So this is a site which is extremely important for containing the infection. Now, if that is so, how can Salmonella perturb the system? How can Salmonella, in fact, dodge or stealthily make a niche in combating so many different strategies? They are transcytosed from the apical to the basal site, and now they are engulfed by macrophages. Now, macrophages in general are an important phagocytic cells, and they would have, in fact, killed anything that gets inside them. But that does not happen. And not only that, they are not able to kill this pathogen, but what they do is, once they get inside, they are able to simply carry this pathogen. So they act as a vehicle. They transmit the bacteria now to these dendritic cells and also to the neutrophils. The neutrophils, rather than killing them, actually does nothing. And on other hand, the Salmonella proliferates or multiplies in these macrophages, and then they disseminate. So instead of becoming a killing cell, they become a vehicle. So imagine the intelligence again. They are now using this macrophage or dendritic cells as a carrier and taking them to the different part of the system to make them systemic. That means they're carrying them to the spleen, they're carrying them to the mesenteric lymph node, they're carrying them to the liver. And that is how the Salmonella is disseminating or spreading. Now in human, you have a very typhi specific serovar, that is Salmonella typhi. Now Salmonella typhi is extremely strict to human as a host. It does not infect any other host which is known till now. And typhoid, you know about the typhoid. You also might have heard about an interesting story of Mary Mallon, who was the first woman to be completely, safe and symptomless, but she spread typhoid to many royal household families, children, and many others. So this is a very stealthy bacteria, and that is why it becomes much more interesting, because they conceal themselves in this so-called vacuole. What is so fascinating about them? Now, when we started working, so way back, I started my lab in 2004. It was a great decision for me to move back to India, and that too in Indian Institute of Science, which is a place for holistic learning. It is a place for deep learning. I have had many students from St. Joseph as a trainee, and all of them not only excelled very well, but they are also having a very different attitude towards life. Not all, sorry, I'm a bit blunt, but yes, two of them they were amazing. And what we started understanding way back with my first graduate student, who is a MBBS doctor and did not go to MD, but pursued his passion to do medicine, a very bright person. Now, currently, he's a faculty in biochemistry department in IISc, Dr. Sandeep Ishwarappa. So we started working on the strategies that the Salmonella may use to somehow combat all the mechanisms that the cell will give. I used a statement that the bacteria have learned very fast about us, but have we about them? Look at this. Now, the moment the Salmonella infects epithelial cells, no? So the red one here is a bacteria. We have many construct, many colored bacteria. This is nothing but a RFP expressing bacteria. We also have a GFP expressing bacteria you see here. We have cherry red expressing bacteria, many different. So lab is, they have an expertise of growing and making a lot of constructs, genetically engineered construct, mutants, etc., etc. So when the bacteria infects the epithelial cell, and epithelial cells are a major producers of antimicrobial peptide. You name it, defensins, alpha defensins, beta defensins, and many other classes of antimicrobial peptide they have, because the epithelial cells do not have a very good burst of reactive oxygen and the reactive nitrogen species like macrophages have. So they rely on the antimicrobial peptide defense strategy, and mostly any bug that enters inside will be killed by these antimicrobial peptide, but not salmonella. And how? So green, what you're staining, that green what you're seeing is a staining for one of these antimicrobial peptide of beta defensin. And now if you see here, now this bacteria, the red bacteria in the inset is getting stained by these green dots. That means that indeed the bacteria is being attacked by the defensin, but they are not killed. How it can be possible that they are being attacked and they are not being killed? So Sandeep went on with this idea that in spite of having a good production of these beta defensin, which would have otherwise killed the bacteria within no time, they are not able to kill. And not only that, they don't get killed, but they multiply. So large extent, they are multiplying in the epithelial cells from, handful of bacteria, say 10⁵ bacteria, they are multiplying to 10⁷. So these kind of fold replication, how can it happen? So he searched and he found out that there is one kind of a transporter, which is known as a H transporter. Now this H transporter does something very interesting. What it does, that it definitely recognizes the antimicrobial peptide, no problem in that, but it acts like a sink. Now, this antimicrobial peptide is taken up by this H transporter. Now, EGF is one of the gene. And what it does further is it chews up this antimicrobial peptide in its periplasmic space and throw them out. Now, when they throw them out, it leads to the generation of the simple amino acids, and other bacteria can use it as a food. So not only that they are trying to curtail their enemy by chewing them up, engulfing them and chewing them up, but also they are providing a food for neighboring bacteria as a simple amino acid. So imagine what can be, how much wonderful can these bacteria go and to what extent by doing this. So EGF is like a sink. So that entire YEJ, EG operon, acts like a sink to take care of the antimicrobial peptide by sinking it in and then chewing them off and nullifying the effect. So cells become just completely clueless that in spite of having such a high amount of antimicrobial peptide, where is it all going? And they all stay safe. So the moment you make a mutation in the EGF, that means you knock off the EGF gene, the bacteria becomes attacked by these antimicrobial peptide and they die. So if you try to compare the replication of wild type between wild type and EGF, you will see that the wild type has grown over 20-fold, but the EGF simply cannot grow and they die. So you see a dip. That means this is an important virulence strategy in the epithelial cells. So yes, the bacteria has entered the epithelial cells and it knows very well how to take care. Now, the bacteria enters the macrophages. Different cell type, immediately it senses that now they are in the macrophage. They switch off the expression of these entire EG operon and they switch on something known as a transcriptional regulator, HRG. Now, this particular gene was discovered by my colleague Amit Lahiri, who is currently a faculty in CDRI Lucknow. And Amit was the first one to identify this gene, so he christened it as HRG. That is a hydrogen peroxide resistant gene. This is a LISA type transcriptional regulator. Now, in the macrophage population, they turn off their EG, they turn on their HRG. Again, a decision factor to understand that now they are in the different cell type. And macrophages being a very strong burst of the reactive oxygen species and the reactive nitrogen species, they immediately express this gene HRG, and they nullify the effect of the reactive oxygen species by simply nullifying them by degrading them. And if you make a mutation in this particular HRG gene, you will see that again the Salmonella succumbs to the oxidative stress. So in macrophages, the HRG gene takes care of the bacteria. In epithelial cells, EGF takes care of the bacteria. And also in the macrophages and dendritic cells, it is the NIR-C. Now NIR-C is a nitrite transporter. Now reactive oxygen species and reactive nitrogen species and their burst are very prominent in macrophages and dendritic cells, but not in the epithelial cells and other cells like endothelial cells or fibroblast. They do produce to some extent, but not as much as they will produce here. And that is a major defense strategy, the first line of the defense strategy in these cells. So again, if you make a mutation in the NIR-C, you see that the nitrosative stress actually kills. Now the red staining, what you see here, is for the peroxynitrite. Now the peroxynitrite is the toxic molecule or the toxic adduct of oxidative and the nitrosative stress. So the chemical formula is ONOO. That is the peroxynitrite, and it is very lethal. It kills the bacteria within few seconds. So if you do an in vitro test and you in a test tube, you generate peroxynitrite and you put your Salmonella, you will see that within no time, within 5 to 10 seconds, they are killed. But within the macrophages or within the dendritic cells, they are wonderfully protected by this NIR-C transporter. So what a transporter does, again, like this sink, they take up all the nitrites or all the species which are floating around so that they don't make this peroxynitrite at all. And the bacteria is safe. But when you knock it out, when you knock out the gene, there is no way, and the peroxynitrite is formed, and you see that the bacteria is getting killed. So different cell lines, epithelial cell, macrophages, dendritic cells, and this also in macrophages, different strategies, different thinking, different decision making. How they can make this decision. Now, one more, again, very interesting take home, message what we can learn from bacteria is how to be judicious with your resources. I always get perplexed and try to take lesson of life from this bacterial behavior. How do they judiciously think and plan their resources? So for example, if the bacteria see that the amino acids are there in abundance in the host, they will never turn on their synthesis gene. Bacteria have the entire machinery to make all the amino acids, everything they can sustain. But if they see that the host has amino acids, say for example, L-arginine, again, Amit hit upon a very, very interesting finding that if they have this L-arginine in abundant, they are not going to take up their or express their synthesis gene. What they will do instead? They are going to only upregulate their transporter, the L-arginine transporter, so they can transport everything that is available near its vicinity. And one must not forget that this bacteria is in a vacuole. That means it is surrounded by a protein or many protein which are derived from the host, and it is secured inside. That means that these bacteria, what they do is, these Salmonella containing vacuoles, they first recruit the cationic amino acid transporter onto their vacuole, and then through that the entire cytosolic L-arginine, they enters the Salmonella containing vacuole. And now from the vacuole into the Salmonella, they upregulate their ARCH-T transporter, and then they get inside the bacteria. So how much ATP they have spent? They have only spent 2 ATP to get their very essential amino acid transported inside the vacuole. If they would have upregulated their synthesis gene, how many ATP they would have utilized? 4 ATP. So by getting this abundant and quenching them, they save 2 ATP. So that is how their skill of judiciously utilizing their ATP And during crisis, they can utilize their or spend their ATP more. But when it is available, never ever. They will always try to have the strategy to quench it from there. An amazing lesson again. And one of the most important hallmark and very, very, it's like a big bang finding of our lab is the salmonella in a single vacuole. Now, a salmonella comes with its vacuole. So you can understand the process of uptake. When the bacteria sees the host cell, it will be taken up. Now, when it is taken up, it will come by default this membrane. This is a plasma membrane of the host. Now, some bacteria like Shigella or Listeria, they would like to come out of this vacuole because they don't like the vacuolar environment. Maybe sometime later, I would tell you about why they don't like this vacuolar environment. But Salmonella, Legionella, Brucella, etc., they love their environment. Salmonella love its environment so much so that in spite of being so stringent and such a less space between this vacuolar membrane and the bacteria, less than a 1 nanometer, the bacterial dimension is somewhere around 1 micrometer to 0.8. And it's a very tight fitting vacuole. So if you see here, this is a green bacteria, and you see here this very tight fitting. Red is the vacuolar staining. So one of the protein, LAMP, lysosomal associated membrane glycoprotein, that marks the vacuole. So it's a very, very tight fitting vacuole. So this tight fitting vacuole, Salmonella loves. But the question is that the bacteria ends up in a vacuole and the bacteria divides. So now the bacteria divides like this and they ends up like this, multiple bacterium in a vacuole. We never found that situation. And that is how when Sandeep banged upon this one vacuole Salmonella theory, it was very interesting to see that vacuolar division happens along with Salmonella division. And how? This is making our life much more challenging for the past 14 years to really understand this vacuolar division. And it indeed is a 1 Salmonella per vacuole, and it is not a multiple Salmonella per vacuole. And this 1 Salmonella per vacuole is the most important and smart evasion strategies, which leads to everything else, like inhibition of antigen presentation, etc., etc. So in a nutshell, which I summarized, in this small one slide, you will be like in some science fiction movie where you see that a journey of a bacteria from the gastrointestinal tract to the different cells, and they are tinkering off of the receptors and sensing the organism, the environment, the different cells, and designing a strategy. It is indeed like this sci-fi movie, and some of you who are interested in making movies, maybe I would love to have you and make few movies on our Salmonella. Right. So indeed, there were too many reports that got reported, but let me tell you, show you something very interesting that this became a cover page of Infection Immunity, where we show that indeed the Salmonella comes in a single vacuole. But the perturbing question here was that there are enough number of lysosomes that are there in the cell. You know about these organelle lysosomes. Now, lysosomes are made to kill the invading pathogen or invading foreign substances. So why are the lysosomes not doing their job when they are there? And lysosomes are toxic granules. They are loaded with cathepsins, they are loaded with hydrolases, they are loaded with degradative enzymes, and they have an acidic pH for its activation. So why is it not killing the Salmonella? What is the reason? That was one of the first questions that we asked. Now, if you see here with a very, very simple microscopic staining, you can stain the lysosomes. All the red here are volume of acidic compartments. They look very pretty, but actually, if I tell you, the system is very simple. You just need to have cells, and there are one chemical, that is the acridin orange. Now, this acridin orange changes, so it's a very beautiful sensor. So when you have the compartment which is acidic, it is going to give you this kind of a red fluorescence. And when the compartment is a basic, it will become green, right? So when you stain your any cells, you can just put some acridin orange. It's a live stain. So you will see that this beautifully, the red compartments are becoming, the acidic compartments are becoming red. So you know that this is the uninfected cell, which is a naive cell, which is not infected with salmonella, has a very wonderful, quite abundant acidic compartments, should do the job. Now, suddenly what happens when the infection is given to the cell? That means when the wild type bacteria, they come in and infect, look at all these red acidic compartments. They disappeared. They all became green. They became kind of non-acidic compartments. Now, the question here is that what is happening to these lysosomes? Are really the volume of acidic compartments are decreasing? Or salmonella is doing something to the pH of the lysosomes and making it alkaline? And remember, all the enzymes work only in acidic pH. They don't work in alkaline pH. So you can have cathepsines and hydrolases loaded, but the moment it becomes alkaline, nothing will work. It is null and void. And it happens. Actually, they perturb this entire process of, having an active lysosome. And maybe this is one of the reasons that this survives so well. Exactly the similar positive control, concanaminine is a drug. Polymycin, concanaminine, they belong to the, the genera of the drug, which acts on these VATPase, and they perturb the pH of the compartments by this proton exchange. So you will see here that these the green, the concanaminine treatment, actually, which is a positive control, also neutralizes the acidity of these compartments, which the bacteria is doing. So indeed, the bacteria plays around, and this is the most important strategy of salmonella to start now multiplying. They have taken care of one arm. They have somehow neutralized the lysosomes. Otherwise, there was no way. Now, let me make you understand step by step how actually the process of uptake and the vacuole generation goes on. Now, if you see the one which is let us start with a non-pathogenic bacteria. By the way, anybody has a question now? Or shall I continue? So if there is no question, so let me continue. And definitely we will take the question in the end. Kindly keep typing in your chat box, because immediately after that, I'm going to read all the questions from the chat box and answer all of them. So kindly do that. So now let me take you to the process of the so-called bacterial uptake or endocytosis and what happens. Now, if you take a non-pathogenic bacteria, say for example, the bacteria that you work with in the lab, DH5 alpha, the E. coli, BL21, or say for example, our common bacteria, lactobacilli, which you eat every day in the yogurt. The bacteria is taken up by the process of endocytosis. And then this is this naive vacuole. This vacuole gets modified by continuously exchanging these molecules or the proteins from early endosome, like EEA1 or the early endosome associated marker 1, transferrin receptor, the RAP5. And not only that, they acquire some of these early endosome marker, but they don't stop here, and they progress and progress and progress. And when they start progressing in non-pathogenic bacteria, they progress and they end up in the lysosome. That means the early endosome to recycling endosome to late endosome to lysosome. And the non-pathogenic bacteria meet with its dead end in the lysosome and they die. So they are cleared off. You have no problem. So that is the reason they are called as non-pathogenic, because they are not able to subvert this pathway. Now, what happens to the pathogenic bacteria? Pathogenic bacteria comes with different flavors. Bacteria, say for example, Shigella and Listeria, they are taken up by the same process, but what happens much before they are about to get hit by lysosome, dramatic things happen. And what is the dramatic thing? That Shigella and Listeria, they have certain secretion system, and they express these secretion systems to produce toxins. And the toxins immediately cleave this vacuolar membrane. They make pores and they come out, they exit. When they exit, they come into the cytosol. And when they exit and come out in the cytosol, lysosome cannot do anything. Why they cannot do anything? Because the lysosome needs another vacuole to fuse with. So it is a dynamic fusion, vacuolar fusion between a lysosomal organelle and a bacteria containing vacuole. So they need those fusion mechanism. And Shigella, Listeria understand this very well, that they don't want the vacuole and they don't want to fuse with the lysosome. So they simply quit. They quit and come. And they are very successful in the cytosol. They multiply. Though they have abundant nutrition and everything in the cytosol, but remember, no life comes simple. As we have a hurdles in the life, it is up and down, what we call it up and down. Bacteria also faces this very, very severely. Now, the up is that they are in the cytosol, but the down is that they are going to be attacked by all the antimicrobial peptides, mostly. And that is what actually happens. So Shigella and Listeria are not something that creates a systemic infection naturally. But now I am not very sure if you have heard certain news. A couple of months back in Kerala, there was a death from Shigella dysentery. But that report is a bit dicey because we do not know whether the death was due to the Shigella. It may be due to something else. Because Shigella and Listeria are not known to cause a very high mortality. They are cytosolic and they do replicate, they do cause a diarrhea, but they are cleared off by the system very fast, and even the antibiotic clears them off very fast. So this is one life. They multiply in the cytosol. Now you have another life of bacteria like Staphylococcus aureus or Mycobacterium. Now, Staphylococcus is a gram-positive class of bacteria. Mycobacterium, as you know, what is a class of Mycobacterium? How do you classify them? They are acid-fast. You do not classify them as a gram-positive or the gram-negative. You classify them as acid-fast. So what these two bacteria, they do is that they are going to get inside, progress, arrest, immediate. Again, absolutely no progressing further to get along with the lysosomes, arrest. How do they arrest? They do certain mechanisms with the help of their virulence protein that this vacuole will never progress into so much so that now they are getting targeted by lysosome. So this is known as a arrest of phagosomal maturation. Now, you know, bacteria always are very interesting because there are more and more research with deep learning is coming up to understand that indeed, is it really a very thin borderline between the so-called vacuolar and the cytosolic pathogen? 10 years back, Mycobacterium's life in the vacuole was completely refuted by a group in Netherlands, Peter Peters group, who published in Cell, saying that the Mycobacterium is actually a vacuolar, not a vacuolar pathogen. It is a cytosolic pathogen. So the cytosolic pathogen, in fact, is there in the cytosol, in the dendritic cells. Anyway, I'm not going to discuss about this. Now, let us see our Salmonella. Salmonella will be taken up and they would modify it in such a way that forget about it's getting into being targeted by the lysosome, but they are going to take every other important and valuable molecule from early endosome and from the recycling endosome and from the late endosome also, but will never get it, allow it to mature in the for the late lysosome. And not only that they will do this, they will multiply very happily within the vacuole. So that is why this is known as a strict vacuolar pathogen or intravacuolar pathogen. So they are here beautifully multiplying. Now, I think that all of you have got this concept very clear about the different fate of the bacteria. So now let us understand that why Salmonella loves its vacuole. If you see here that in the epithelial cells, they are taken up and then they will use certain virulence factors and they are going to multiply. Now, don't get perturbed by all these signaling molecules, type 3 signaling molecules, etc., etc. The only one message I would like to tell you is that this particular, diagram now is getting remodified, because you'd never see multiple bacteria in a vacuole until and unless the bacteria has a problem. It will be only a single bacterium like this in a vacuole. And then it makes these some beautiful filaments known as a SIF, salmonella-induced filament, where they will use this filament to keep these salmonella-containing vacuole tethered together or held in one place. So they make these wonderful cell structures known as a SIF. Now let us peer into the cell, and now you are convinced without doubt that you know where do they live. So they live in vacuole. Now this is their home. This is a macrophage raw 264.7 cells which are infected with salmonella, and they live within their home. And this home is nothing but the salmonella containing vacuole. RAP7 is one of the marker for the salmonella containing vacuole, and green is the GFP expressing salmonella. And you see here that they have plenty of home to live. They have plenty of home for replication. And you will also notice one point here, that there is absolutely no way by which 2 salmonella ends up in a single vacuole. So see, this is the home. Each one beautifully laced by this single vacuole. So it is a single salmonella per vacuole. So that is where they live. This vacuole is made up of more than 40 odd proteins. So we are actually understanding in great detail about this vacuole. We have a way to isolate this salmonella containing vacuole and do a whole lot of study on these vacuole. Very fascinating compartments. So the question is, definitely it is a single bacterium per vacuole. But how are they in fact dividing, and is it really true, or it is something which is specific to certain cells? So my colleagues Sandeep and followed by the Belina, etc., they started looking into with the help of electron microscopy, because electron microscopy, nothing can beat the resolution of electron microscopy. See, you will always have a issue of resolution with any other form of microscope, even confocal microscopy. Super resolution is better, but electron microscopy, there is no questions asked, though this is a very tedious technique. So when they started looking into this with the help of electron microscopy, you will see that very beautifully, a very tedious experiment this was, because you need to really capture more than 1,000 fields to understand that, and very thin sections are required, and it's a very difficult experiment though, but a very powerful tool. So see here, the bacteria is dividing, a bacterium is dividing into 2. So this black is this bacteria which is dividing into 2, and the white, what you see here, is the vacuole. See, this is this bacterium and in a vacuole. You may ask a question that why this is looking round, because these are rod-shaped bacteria. This is just because the sections that you have cut, So if you are cutting it in a section, then you will only see this round section, like this, the plane of the section that is observed in the image. So this vacuole is undergoing invagination. Very fascinating. Bacterial division, all of you know, I am sure. They use diviso machinery triggered by the FTS complex, FTSZ complex. There are again 14 to 15 odd protein, and they mark your mid-cell division site, and they divide. We understand very nicely, we understand. But what marks this site? That is the vacuole, and the vacuole is marked just with the, as if it is somehow timed in such a precise manner with bacterial division. And they divide into 2. Now this is the complete division, what you are seeing here, one of the event, the complete division into 2, and it divided. So you see here a complete division into 2. This is undergoing the division, and then you see that everywhere that you see, it is a single bacterium per vacuole, and they end up in a single bacterium per vacuole. Now the interesting part is that the single bacterium per vacuole is there. Definitely it is there, but how? That is the question. But how? I think I need to admit there are 2 people who are waiting to be admitted, Okay, 1 minute, because I don't want anybody. So I will say admit all so that everybody should be there. Okay, fine. I have pressed admit all. One person is again waiting. So this is also admit all. So hopefully everybody got admitted now. Okay, fine. I will do it from time to time. So indeed, in the cell it happens. But is it only with the cell? Because raw macrophages are macrophage monocyte-like cell line and you are isolating them. So anything can happen. What happens in the physiological level in organ? So try to have a very closer look in, again, one of the important cell line, which is the intestinal epithelial cell line. Again, you see that it's a single bacterium per vacuole. And now you go to liver section. Now in the liver, I'm admitting them, but somehow they are not getting admitted anyway. I hope that they will be able to see and the YouTube and get benefited. Now the liver, when you use an animal model, and we are very lucky because a mouse is an animal model for salmonella. This is a dead end model. The mouse will die within 8 days of salmonella infection. They mimic the typhoid-like symptom in the human. So they are the best model that we can have. It goes on from the gut and it spreads to the liver. It spreads everywhere, spleen, mesenteric lymph node, and to the liver. And when we took these liver sections, you will see that even in the liver, a physiological organ, a very important organ where salmonella harbors, it is a single bacterium per vacuole. See, single bacterium per vacuole. Division, undergoing division. We'll get into single bacterium per vacuole. Such a sophisticated and a highly intelligent process happening in an enclosed environment and a highly architectured tissue of a liver. It's happening. It's happening in the cell line. It's happening in the liver, which is a very important organ. So the strategy of single bacterium per vacuole is indeed a strategy that takes salmonella a way forward than any other bacterium that I can think of. And now you know, and I think that this, whatever couple of minutes that we have discussed, it will make you convinced that do we now really know enough as they know about us? Do we know about them? That always remains a question. Now again the question comes, why do they love their studio apartments? It is so stringent. Why they should love it? Some of us, of course, love the studio apartment because there are many reasons for it. You don't have to clean so much, and you don't have to worry so much about anything. You have everything at your hand's length. And I lived in one such studio apartment in Germany, and it was really extremely fruitful because I did not have even a single minute to take care of a big house, etc. Because 3 hours I was in the lab, hardly 1 hour in the room. So that doesn't make sense. So I love my studio apartment. But why bacteria would love their studio apartment? Because if they would have had a spacious phagosome, they could have acquired a lot of nutrition, etc., etc. But no, they love it. They love it because they are single in that studio apartment. Of course, in the studio apartment, if you cramp it up, there is no way. You can't live. So they love it because they are single in the studio apartment. They have very reduced competition for the nutrient. They have increased surface area for the nutrient acquisition. They also have a very enhanced surface area for each bacterium to secrete the effectors into the host cell. And then what happened to the host? Now the host needs to start thinking, oh my God, these studio apartments and with the bacteria are increasing in number and occupying us. How do we strategize to attack the individual vacuoles? How do we strategize to attack the individual salmonella? Then they have to start thinking, and the thinking comes very late. That is what, do our 30,000 genes make them think faster than bacteria? The answer is no, because they are far more smarter. But of course, we are what we are, and we are going to keep getting fascinated by their strategies and take a big lesson from them so that we know what to do when crisis comes. So that is how they love it. But though they love it, how do they divide? My colleagues, Sandeep, Debalina, and many others, but started by Dr. Sandeep, they took a very important lesson from one of the division machinery, that is the mitochondria. Now, you all know that the mitochondria is nothing but an endosymbiont. One of the best example that I can tell you, which is an amazing example of alpha proteobacteria getting inside the host and becoming such a harmony with the system. The cell did not react, the bacteria did not react, and they came to an harmony, and it became one of the important organelle, that is a mitochondria. Now, the mitochondria is also double membrane. So the outer membrane division happens with the help of this dynein or the dynamine related protein and FIS1. And the inner membrane division, it happens with which is a homologue of this bacterial division machinery, that is FTSZ, which is known as the MGM1. Now this outer division machinery and inner division machinery in mitochondria, they also very beautifully coincide with each other. And that is how you get mitochondrial division. I'm sure all of you know about how the mitochondria is a very separate entity with its own genetic material, and how do they increase their number by mitochondrial fission. So they divide. Now, how do they divide? The site is marked by DRP1. And when it is marked, these are all motor proteins. With the help of GTP as a source of the energy, they in fact undergo this so-called fission. And one mitochondria becomes 2. One mitochondrion becomes 2. So that is how the mitochondrial number is maintained. Now, my very bright young colleagues, and that is why I always say that indeed I am always very, very lucky to get the brightest mind in my lab. That is my star. And if I talk about holistic learning, I do believe that there is something much, much more that science cannot tell us, that why do I get the luck of attracting all the bright minds? And that is what has happened. So they took the inspiration from this mitochondrial division machinery, and they asked a question. What if this salmonella also utilizes this division machinery to divide their vacuole? And if you perturb that machinery, then what happens to the salmonella? Now let us go one by one. If you look at this salmonella, this is a control where your machinery, everything is intact. And the salmonella, green salmonella, and the red is a vacuole marker. This time it is a LAMP1, lysosomal associated membrane glycoprotein, which is a very important marker for SCV. Now the salmonella is dividing beautifully, all one in a vacuole. Very nice. The moment you perturb that division machinery with the help of sodium orthovanadate, which is in general protease inhibitor, but if you use at this particular concentration of 100 micromole, then it specifically inhibits to a greater extent the DRP1, that is a dynein related protein. Now, if you inhibit the DRP1, see what is happening. All the salmonella are entered in a single vacuole. So you see multiple salmonella in a given vacuole. Okay. So instead of one salmonella per vacuole, now they ended in multiple salmonella per vacuole. Now the question is it again happening in all the cell types? Yes, it happens in all the cell types. Macrophages, a single bacterium per vacuole. Epithelial cells, again, you beautifully see loaded with salmonella, single bacterium per vacuole every time. But if you perturb the division machinery, they become a vacuole containing many salmonella. So we term it as a VCMS, vacuole containing many salmonella. Now, what happens? Now, see, if you see here, the salmonella divided up to certain extent in a given vacuole, right? The question is, if that is so, can the vacuole become bigger and bigger like a balloon? And they can keep on dividing. Can that happen? But you know, that is not the case. The moment you perturb this vacuole, vacuolar division, something happens to the life of salmonella. Let me tell you what happens. Now, the sodium orthovanadate here is the chemical agent which is killing your DRP1, right? That is the only thing you need to say. Now, the DRP1 is something which is very important for the division of the salmonella containing vacuole. So if you use the DRP1 inhibitor, that is a sodium orthovenodate, you see that your VCMS numbers percentage is very high. Just concentrate on this. In control, it is single bacterium, it's very low. But in SOV treated cells, they are very, very high. Almost more than 60% are in VCMS. But what happens to their life? That means what happens to their intracellular proliferation? So if you look into the fold intracellular multiplication within the cell, it is just the opposite. The control ones, they replicate to more than 8 to 10 folds, but the orthovenodate treated cells, the replication drastically falls. What does that mean? That means that if you perturb the home or the niche of salmonella, making them to come under many salmonella in one vacuole, then immediately their level number falls. So one vacuole per strategy is the most important strategy and the most hallmark novel finding of our lab, that is keep us going and keep us, making us more and more challenging to ask these questions. So actually our life revolves around a vacuole, and that is what is the beauty of the system. So you know now that you cannot change the niche. If you change the niche, they will grow, they look beautiful, they grow, but in all their number falls. So the one vacuole per strategy is the most important thing. And then what, how it is getting marked, the site of the division, because somebody has to come and mark it and completely align it with the bacterial division. So the bacterial division is by FTSZ, but the vacuolar division is by the DRP1, but still somebody has to make it, align it. So in fact, endoplasmic reticulum does this job of aligning it and making it a mark. So Debolina, who is currently doing her postdoc in Emory University, USA, she figured out that it is indeed the endoplasmic reticulum which marks the site. Then the DRP1 also gets recruited, and then this division takes place. Yeah, so she isolated this. These are isolated SCVs. If you see here, these are all isolated SCVs. So what you do is you take a cell, any cell, epithelial cells, macrophages, you put bacteria, it gets infected, and then you have a way to isolate them. You isolate them, and when you stain for this DRP1 on those isolated SCVs, you see that yes, indeed, they are with LAMP1, they are with DRP1, and they become very beautifully co-localized, saying that yes, you can get this division done with the help of this DRP1. So electron microscopic studies also suggested the same, that the dynamine related protein is the most important thing. So a lot of other, studies were done where the ER signatures, KDL, etc., was used to indeed do some live imaging to figure out that how does it mark, that why ER is and how ER is marking the site for division, vacuolar division. So if you see here, these are the bacteria, and you see this interspersed ER. So this is how this ER, look here, this is the one. So this is what the ER is wrapping around. This is very, resolution of the confocal is very low. So that is how we have to do the EM, more EM, to figure out that how it is actually getting marked. So you do have salmonella proteins which play a very important role in the division, SCV division. It is just not coming only from the host. It is also coming from the bacteria, which is maintaining this division. And the lab also figured out, in fact, Debolina only figured out that what is that protein which will allow it to divide and keep the single bacterium per vacuole. So if you see here that this is, when you do not have this particular protein, RFAG, they come under multiple bacterium per vacuole, and this particular bacterial mutant, that is delta RFAG, is completely non-virulent. They die off. Even the mouse, if you inject them or infect them or feed them with this bacteria, they don't die. But if you feed them with wild type, they die within 8 days. So from every angle, it is very important to see that indeed they are getting disturbed or degraded. Now, you know that now my bacteria is in a vacuole. So very quickly, I will tell you that if my bacteria is in a vacuole, how do I make them, what is my intervention strategies? Because all the antibiotics that I'm giving may not be even reaching or crossing the vacuolar membrane. or the barrier. So only when the bacteria is in the bloodstream, we will be able to tackle it, but not when. So with our excellent collaborators from material science, Professor Ashok Rai Chur, we figured out a way to deliver the antibiotic ciprofloxacin in a nano carrier delivery, which is a mesoporous silica nanomaterial. And that delivery, in fact, helped a lot. What happens when you deliver? So your CIP-L means ciprofloxacin. CIP is ciprofloxacin. If you put it in the MSN, that is a mesoporous silica nanomaterials, it will be CIP-L MSN. And CIP is alone, CIP in PPS or in water. And MSN is the pure only nanoparticle, unloaded nanoparticle. And this is the mouse experiment, which is very, very clearly indicating that if you have the ciprofloxacin, which is loaded in the mesoporous nanoparticle, only half of the dose, that is 10 mg per kg body weight, half of the dose is able to make all 100% of the mice survive. But the same half dose, that is 10 mg per kg body weight, if you use Cipro alone, they die. Okay? And if you use 20 mg Cipro, double the dose alone, then only the mouse survive. Now you understood the meaning of carrier molecule, that not only that you decrease your concentration of antibiotic, but you also make sure that the bacteria in future will not become antibiotic resistant. And understanding this vacuole is very important because you are delivering it right. You are targeting it right. Otherwise, you will dump them up with many things. Okay. So I want to have a lot of question answer sessions. So what I'm going to do is I will just come to an end. That there are many, many things, exciting things that are going on in the lab. And as I mentioned to you, plant also are not spared from Salmonella. So we know the trans kingdom invasion, that Salmonella gets systemic in the plant, especially tomatoes and other salad vegetables. So all of my young colleagues, be careful when you eat salad vegetables. It's not a scary thing, but it's always better to eat cooked food. And the same tomatoes, if we feed to the mouse, they become systemic, and then their feces, they shed Salmonella, and then the feces mixed with soil, that is how their burden increase. And on the other hand, they make these so-called biofilms or the microcolonies. They're very sturdy, tedious, so no antibiotic can penetrate. And they suppress antigen presentation, and everything is happening because they are in single bacterium per vacuole. As I mentioned to you, said that these are all my stars, and there are many, many stars which have passed by the lab, all the summer trainees, all the project assistants, etc., who are now a vital force, vital contact people who are making their own research and own life, and their, very, very important contribution to understanding and the bug, which I still feel that have we understood enough. So my thanks to everybody, thanks to my collaborators, and finally, thanks to you and for you. Definitely there is something which is very incredible that is waiting to be known. So venture it out, and you are always welcome to, talk to me, talk your scientific ideas, get opinions, and it is a very wonderful discussion. So what I'm going to do is I'm going to stop my slide here, and then we will take up the questions. Thank you very, very much for listening. And I hope that these online session, we were able to still connect, because I do feel that learning happens, it carries, it's a form of a wave. So what my feeling and my passion to tell you should get across to you on the other side of the table. Though we are not seeing each other eye to eye, but I know that I am able to make the contact. So now I will be very, very happy to take up questions. Okay, so now there is one question from Dr. Atul Dayalan. What is the CFU of Salmonella infection to SCV? Now, the colony forming unit that we use for infecting the cells or the mouse is somewhere in the range of 1 × 10⁴ to 1 × 10⁷. So this is the CFU that we use. In case of cell line, or when you infect, you use the term MOI, or the multiplicity of infection. That means MOI of 10. Normally we use MOI of 10. That means it's a 10 bacteria to one cell. So that is how you can vary your MOI and you can put up the experiment. So, but in case of the animal, it will be 10⁷ to 10⁸ CFU per mouse that you use to infect your animal. The interesting part here is that though you calculate your MOI 10, that means you are giving 10 bacteria to one cell, is really those 10 bacteria entering inside the cell or getting internalized? Actually, it's a very difficult question, and we are working a lot on this to understand that what is the signaling mechanism which makes the bacteria take up cell, and if that is so, how many numbers? So we have a lot of mathematicians with us to, in fact, help us with some mathematical modeling to understand that. Because if you overload the cells with bacteria, are they going to take up all, or are they going to only take a some and then discard the others? So there are a lot of these kind of the questions. Thank you, Dr. Atul. Kindly ask your questions. See, we are not face to face here, so you are free to ask. You should not be feeling shy.
Excuse me.
Yeah, hello.
Myself, Dr. Pankaj Kumar Singh from IIT VHU.
Yeah, hi Pankaj, hello.
Your presentation was very interesting regarding the salmonella.
Thank you.
You have presented very beautifully the story about the salmonella smartness. My question is that salmonella is occurring in the vacuole. That is okay.
Yeah.
That's the problem behind its cause, like the salmonella typhosa, that typhoid causing, I think.
Yeah.
Exactly. But which toxin is released by the salmonella, and when it go on to release? Can you say something about that?
Sure, I'll tell you. So it's a very good question. Now, salmonella is having pathogenicity islands. Okay, so these are known as the virulence factors. Now, there are two pathogenicity islands, that is the pathogenicity island 1 and pathogenicity island 2, which is there in salmonella. Now, these two pathogenicity islands are horizontally acquired pathogenicity islands. They're a big chunk of DNA, around 40 kb long DNA. They come with their own operon, their structural protein, their secretion system, and the effector proteins. And the way by which salmonella enters through into the cell is by making these injectosome and secreting the effector proteins. And that is how they are kind of endocytosed. Once they are inside and they are in a vacuole, they take help of SPY2, another pathogenicity island, and they make a translocon. I did not get a chance to tell you that how the translocon looks like when it is in the epithelial cells. Sorry, in the macrophages. Within the cell, once this injection needle system is made, they start making these virulence factors, certain toxins, certain modifying agent right into the host cytosol through this translocation system. There are more than 40 odd proteins which are secreted by the salmonella. Now, there are a couple of groups which also reported about these certain salmonella-induced toxins which can create a very subtle pores into the vacuolar membrane. And under certain conditions, they do this because when the threat is very high, that kind of a very, very small pore give an indication of a damage to the vacuolar membrane. And then what happens is it gets recruited by a certain autophagosome or autophagic membrane. Instead of getting into autophagolysosomal pathway, they sometimes even hijack that and become again a very secure autophagosome where they replicate. So the entire secretion of these salmonella proteins or the toxins is via the secretion system, which is very timed. So within the bacteria, with the help of secretion system, pathogenicity island 2 and a couple of other secretion systems, they are going to secrete these proteins. Dr. Pankaj? Hello?
Excuse me, madam.
Yeah.
CDT. SCDT. Can you say something about that?
STDE?
SCDT.
SCDT?
Yes.
Oh, that is the cytolethal distancing toxin you're talking about?
Yes.
Yeah, so these toxins, they are also not only secreted by Salmonella, but they are also secreted by Shigella group of proteins. Yeah. When Salmonella infects host cell, this toxin, in fact, is named as a distancing toxin because it creates some kind of a destruction to the epithelial cell. Much before, much before, say, in 1980s and all, there was too much of work on these cytolethal distancing toxin. But then it was figured out that this is one of the most minor toxin that can be causing a rupture to the cells. And there are now, that is how the work on the toxins of the Salmonella gained less and less importance. And the work on the other proteins, because in spite of having any kind of perturbation factors, they take care of the yeah. Okay, one more thing. Very good question about CYP-B. Now, CYP-B is, again, an important virulence factor or the proteins, and it induces the pyroptosis in the macrophages. Is there any drug which destructs or changes the orientation of the protein? This is from Hassan. Now, CYP-B is one of the most important virulence protein, which is secreted by Salmonella pathogenicity island. All these studies which are reported, mostly you will notice that they are done in the single isolated cells, and this pyroptosis paper which came out that CYP-B induces pyroptosis. Not only pyroptosis, there are many other modifications that it induces. That was known to, in fact, perturb the entire maybe process of pathogenicity. The same thing could not be replicated when you have the whole host as the host when the Salmonella gets in. Now the question is there any drug which will destruct or change the orientation of this protein? The answer is no. Till now, though there is a lot of work which is getting done by Stanford and by Karolinska Institute, one is a crystallography lab and another is a biochemistry protein lab, where they are trying to understand a certain protein or manufacturing of the small molecules which will either bind to the effector proteins or it will bind to the base of the translocon system to make it null and void. Some of the molecules are in the pipeline, but no, not yet. Not yet, Hassan. Till now, we do not have a drug which is going to destroy this or change the orientation of the protein. And if somebody can, this will be really very helpful. But having said that, we need to take care that they comes with a layers. So you have to make your strategy to deliver it into the compartment. Yeah, very nice question. Okay. There are no more questions?
Andrea, tell me.
Hello?
Ma'am, I think there are not many more questions, ma'am.
Okay. Yeah, if there are no more questions, then let me thank all of you for for this wonderful few questions. And in case you have any questions, please feel free and keep putting it in the chat box. I am going to be coming again tomorrow, and I can indeed answer all the questions which might have come up in your mind. Yeah. So thank you very much. Thank you, Grace. Thank you, Dr. Madappa. And thanks to all of you, all my young colleagues, my precious teachers. Thanks. Over to you, Grace.
Thank you so much, ma'am. Thank you.
Thank you so much, ma'am, for this enlightening session. I'm sure all of us learned a lot from you. And I would now request Sri Lakshmi to proceed with the introduction for Dr. Harinikumar.
Good morning, everyone. This is Sri Lakshmi here. I would like to introduce Dr. K.M. Harinikumar. Dr. K.M. Harinikumar is presently working as professor at Department of Plant Biotechnology, University of Agricultural Sciences, Bangalore, coordinator for DBT HRD program, coordinator for Agri Innovation Centers at University of Agricultural Science, Bangalore. He completed his graduation and post-graduation at University of Agricultural Sciences, Bangalore. International post-graduation course on adapting to the climate change, biotechnology in agriculture in a world of global environmental changes, held at Rehovot campus, Hebrew University of Jerusalem, Israel. He started his career as instructor at University of Agricultural Sciences, Dharwad. Later, he joined Mysore Sugar Company Limited as cane officer, then as chief cane department officer at Pandavpur, Shakkara Shakkare Karkhane Limited. He gained a huge experience in converting huge potentiality of sugarcane into bioethanol. His major research area of fuel biotechnology and microalgae for biodiesel production. He got 8 research projects on biodiesel from state and central government as principal investigator and also served as co-principal investigator more than 3 projects. Full-length research article published in Referred Journal 78, published 4 books. He attended national and international conference, seminar and workshop and presented 54 abstracts. He attended 18 training programs and he organized 21 national and international conference and training programs. He delivered 8 talks in television program and 12 in radio program. He is the member of more than 13 professional bodies and visited Germany, Israel, Thailand, Japan, and South Korea for scientific interaction. And he has also got 4 national and international awards. Handing over to you, Dr. K.M. Harinikumar. Thank you.
Good morning. I think you are nearby. Good afternoon to all, and thank you for Gracie Prabhakar and Sri Lakshmi for brief introduction about this. Okay. Hello, audible?
Yes, sir, you're audible.
Yeah, slides are visible?
Yes, sir, it's visible.
Okay, now I'll start that our lecture is mainly on biofuels, the future renewable energy. So all of you know that energy is come from food for us and also
so what I'm saying is that whenever we kind of today's session, I'm making my list now. It's kind of presented at the end.
Can you mute? Sri Lakshmi, can you mute others or?
Yes, sir, I have muted.
Yeah, okay.
You can continue, sir. Sorry for the disturb.
Okay. Slide is not moving. Yeah, okay. That is the fuel of the future. Probably all of you know that the position of the fuel. So what is the requirement of energy? World energy requirement. It is a quadrillion BTU. That is 1 quadrillion is equal to 1,000 trillion. So much of the requirement we need it. And there's something is displaying. Can you remove that? Please move this window away from the shade. Something is displaying. Yeah. Okay. Now. So good. I think we have a lot of requirements. Look at this graph, how it is. There's a lot of demand for the petroleum and other liquids and also natural gas. The coal requirement will be declining because of the deposit will be over. And the entire world is not looking to use this coal as a main source of that fuel because of the lot of pollution. So that is another reason. So the renewable energy is also increasing. So 2040 it is reaching around more than 125 quadrillion BTU. This is a British thermal unit. Is a unit to measure the energy requirement. And nuclear is also requirement is around 50. So this is the requirement of the fuel for the next generation. So why if you come to the
Sir, 1 minute. Your screen has not shared, sir.
now?
No, sir. It has stopped sharing.
Now?
Yes, sir.
Okay. There's an energy demand when you come to the fuel mix. There are different sources like oil, nuclear, hydro, and gas and coal. Coal is around for 20%. I think another 20% from the major is a gas, hydro, and nuclear. But look at this. There is a graph declinement. So the coal, you have to reduce the oil requirement in future because the deposit is lower, and also it creates a lot of pollutions and also greenhouse gas effect. So these are some of the reasons. And look at the renewable energy will be increasing in 2030. We'll get a more of renewable energy than compared to that. This is what the trend is there already. We need. This is the reason why the next fuel, biofuels, is the future energy requirement.
Sir, 1 minute. Can you please share it full screen, sir?
I'll do it.
Thank you, sir.
Okay. So now let us go for the global primary energy consumption year-wise. So the major consumers are the Asia. So we need to look at that. The major consumer is Asia. So later it can be OECD, that is Organization for Economic Cooperation Development, include UK and some of the European continents. Countries are included in this. There are 37 countries which are included in that. So that means Asia, we are in the group of Asia, that is called non-OECD group also, including Africa, America, and Middle East. So Asia needs lot of fuel. So how to run that fuel? There is a requirement of our energy requirement by 2050. So this is the way how the biofuel are getting from the biomethane, biodiesel, or bioethanol. So look at this graph, you can see the carbon footprints. So it can increase it. So in 2045. So even now we are in the 2020. So it started now. So this the demand for biofuel will be increasing. So it is enormous. I think it can go beyond the 0.6. There is a demand when you reach the 2050. That means the ethanol or methane, diesel from different sources. I'm telling here the different sources. So out of that, methane from corn and other things, or biodiesel from the genetically modified crops, and from cellulosic crop, ethanol, bioethanol, sugar beet and corn, and also biojet. Vegetables oil, maybe algal oil also, biojet fuels, and biodiesel from rapeseeds and soybean bioethanol. So like that, there are a lot of sources available. We'll come back to you when you go to the next slide. So what are the source of types of biofuels? How we are going to get the biofuels? So why it is that? Another major reason the biofuel requirement is that there is a deficiting the fossil fuel depletion. So in future, for example, we have a vehicle, but without petrol, diesel, how to run it? So that is the major question for the next generation. So look at the 2046, where you cannot get the coal at all for in particular. It may be depletion is there. The chance of getting is very costly, not able to affordable to purchase that coal. So similarly, the oil is also 2046. So like we have a gas is 2046, but the coal will be there, but already people are not using coal much because of the low quality or low energy coal which is available only now. So that required a lot of pollution and also causes a lot of pollution by emitting carbon dioxide and carbon monoxide. So this is the way. So when you know oil, no natural gas and no coal, what is the source of energy? Today, what you're getting the electricity is mainly from the coal also. Apart from the hydro project, we also have a thermal project, mainly they are using the coal. In future, without coal, so we cannot get electricity. So natural gas is another way it can produce electricity. And I know all of you know that every kitchen now have a natural gas. Just imagine without a gas, in the morning when you get up without gas, how to cook your food. And oil is there, but also petrol and diesel, how to go to office or how to move from one place to other place without run. This is the really critical position. So with the fossil fuel depletion is there in the future. So you should think alternative now only. That's the reason why biofuel will be the future energy. Look at these children going to the school when there is no petrol and diesel. So this may be the situation if you not now concentrate on biofuel production, if you know biofuel production. So the people, students has to go, or probably the youth you are there now, your children has to go like this into the school when you reach 2040 or 46. You can call this as a 2040 vehicles of 2040. We have to use only bullock cart, horse cart to run and move around. That was really the age as seen earlier. People are using it. The same way you have to use back. Again, you have to come back. The old histories come back again. So this will be the situation when you not get the fuels. Let us come to the what is biofuel. Probably all of you know that already. When you come to the biofuel, there are three categories. Solid, that is in the biomass. Liquid, that is called bioethanol or biodiesel. Gaseous fuels, hydrogen and also methane. Consisting of derived from the biomass. Biofuel always, all the fuels which are derived from the biomass, particularly either living organism or their metabolic byproduct, or such as manure from the cows. So this is how the biofuel is type of fuel made from the organic material produced by living things. In contrast to the fossil fuel such as coal, petroleum, or a natural gas that come from a long dead plant and the micron. These are deposit actually. You're taking a deposit from that. Now it is depleting. The deposit will be depleted in 2040 or maybe 50. India will not have a sufficient energy required energy will be available for us. So the next one is biofuel includes any solid. I form is there. It is from either domestic or agriculture waste and other. The fuel does not add to the stock of total carbon dioxide in atmosphere. So that is a very important concept where the biofuel is a type of fuel that does not add a stock to the total carbon. That is our major concern. When the carbon dioxide increases in the atmosphere, what happens? The temperature increases. So that affect on all global, that is called global warming. So all of you probably, if you are experienced, when you go to a community hall or when you go to a classroom, just close the doors, close the fan, close the AC, just sit for 30 minutes, we'll get a warm feeling. Just we cannot leave in half an hour or 1 hour in the classroom without any cooling or maybe without it. So this is how if the entire earth happens, what happens? So what will be the concentration? This is the emission is one of the major problem. Come to that. I will come to you and discuss your details about that. So what is the carbon dioxide level presently? What will be the final at which we cannot live at all? So if we come to the country-wise, world biofuel production during 2020, the major is US. US producing the bioethanol using the corn, that is transgenic corn, herbicide resistance corn. They convert the entire corn into fuel ethanol. So what they did, they take the corn, divide into sugars, carbohydrate, and divide into a protein or the lipid. So the protein or lipid will go as a feed to the animals or poultry, whatever, birds, and whatever the sugar is converted entirely to the ethanol and farmers selling in the form of ethanol. So when I was a student in this campus, when I'm doing the master, during that time, the US never produced bioethanol. Now today, it is #1 in the world. That time, Brazil, India, only few countries have production of ethanol, particularly they're doing it now today, because it is take a lead to produce the biofuel in the world. The next place, go to Brazil. Brazil, there's a lot of vehicle running only on bioethanol and also flexi engine available in the country. They can use both petrol and diesel. European Union using a lot of diesel. The major source of the biodiesel we used in the European Union, there are buses, vehicle run on that. The next is China. So we have you are very meager. You are coming in the just 3%, not even 1%. Other countries, less than 1% in India are using the biofuel when you compare to the world. That means there's a lot of scope to produce biofuel in India And also a lot of job opportunities, employment will be generated out of this. Then come to the India, come to the Indian scenario in that ethanol demand and blending rate. Actually, the government of India already approved for 20% blending without modification of the engine. But presently, we not reached even 5%. So look at that, recently we are reached the 5% of blending. The petrol, whatever you are taking now, it is blended with 5% of ethanol. So why it is not, but the government has 20% now because of the availability of ethanol. Ethanol mainly used for the production of liquors. The second goes to pharmaceutical industries. Then it also go to the cough syrup, for example. The next is go to painting industry. And so many industries required ethanol. So there is a good solvent, So, but now there's a reason why not able to reach our own target at all, even 20%. We are in the less around 5%. So that means there's a lot of demand on the ethanol. And also you look at that, around billion liter, 2.5 billion liters has been used for that. So to reach 10%, we need 5 billion. To reach the 20%, we need a 10 billion ethanol. But we are not able to. There's a lot of scope also. Now government is taking initiation to produce bioethanol. So this is the biodiesel market size and used by in 2015 and 30, what will be the projections. So the main source of biodiesel now, they're used for the transportation in railway and buses are using these biodiesels. And the second one is construction and mining. They are also required now using the biodiesel for construction and mining. Next one is the power generation. So we can use the biodiesel generator, can we use this biodiesel and can generate the electricity, and other source is that so requirement for them. This is the statistics telling you how we can use, and using the biodiesel is also very important for us. We come back to do what is the biodiesel. Come to the types of biofuels based on its uses. The first one is bioethanol. Bioethanol is essentially pure alcohol, and it is the most ubiquitous of the alternative biofuel making their way into people's gas tanks. It can be made from various sources, starting from a cellulose. Also, you can use to ethanol made from the sugarcane bagasse, waste product from sugar processing, molasses, and also can be done directly from sugarcane juice, and also can use in the corn. I told you in the success story of America, where they are converting entire corn into a bioethanol and also animal feed. Some of the advantage of it is a renewable. That is what we need. As long as sun is there, we can grow the crop and get the ethanol. And regenerated given sufficient crop yield. Domestic production reduce dependency on foreign fossil fuels and boost rural economy. That is a very important for us. Even though whatever the cost is not the matter, here even the less cost is there actually. So only you have to reduce the dependency on foreign fossil fuels and boost ethanol. And very another important, ethanol is a cleaner burning than the gasoline. It releasing only 50% less greenhouse gas emission compared to the petrol. Gasoline refers to the petrol. Even in America, the entire thing is mixed with the 10% blending. They made a compulsory. Whatever the ethanol produced out of the corn, they're blending. So MTB is another one compound which actually a carcinogenic. It is a mixed in the petrol to enhance the octane number or enhance the burning capacity of the petrol. So where it is a carcinogen, but it is banned in America. Now in people now in India also they like to ban it or maybe completely have it. So this is an advantage. Even blending with the petrol has giving a lot of benefit to the environment and also cost also reduction. The advanced cellulose ethanol can make ethanol fuel from waste. Maybe that is another way. Even if you look at the south of the country like Sweden, during my visit, I visited the unit. Even though it is not economically feasible, they start producing ethanol using the wood waste or wood powder. So whatever they're using, they're not able to grow any crops like us, but still they have started the work with wood matter or cellulosic ethanol. There are people assume that ethanol derived from the corn, beet, sugarcane compete directly with the food supply, drive the cost of other foods and the kind. No, it is not actually the correct. So what am my feeling? I worked as a farmer. Basically, I'm a farmer and also worked in the sugar industry more than 10, 15 years. So my experience that, so there's a lot of gap in that. If I take, for example, sugarcane, where the Brazil is mainly producing ethanol using the sugarcane itself. When the sugar price drop down in the international market, they're going to produce ethanol and blending with the petrol, or we can also have a neat ethanol engine available in the country. They can use it also. So now they're using it in a way. So similarly, India can also do. When you come to the sugarcane yield, so the average sugarcane yield is around 35 ton per hectare. But the potentiality, it is there up to 60 to 70 ton. So why not we can produce ethanol using the sugarcane juice? Or recently the government of India taking initiative to produce. It never compete. When the farmers get a good price, they certainly they grow. Even the corn, sometimes nobody is to purchase from the farm. So this is the reason why. So there's a lot of potentiality. People have a less knowledge, or maybe may not know the productivity of the sugar, productivity of the cane, or productivity of the corn, or the, what is there in that productivity. There are two concepts. One is the actual production, and what is the productivity. So there is a productivity we can increase. When you give a proper price to farmers, we can do. That is not a problem. Come to the ethanol as less stored energy, no doubt in that. We can mix with some other things where we can also mileage will be less, but the cost is also very less. The 1 liter costs you around 32 rupees, whereas petrol is more than 80 rupees. It is currently require more energy to create large quantities of ethanol than it produces, but it is not correct. However, cellulose ethanol may be a magic bullet to solve this problem and food competition. Converting a standard gasoline engine to run high blends of pure ethanol can be costly, but not required for present at present. We can at least go for the blending up to 20% without any modifications. The second important one is biodiesel. It gaining a very popularity. Biodiesel mimics petroleum waste diesel fuel is derived from the vegetable or animal oil. You know, the first vehicle, the diesel vehicle is actually name of the scientist who discovered the vehicle used, the first vehicle or the engine used to run is used, they use the grounded oil to run the engine. So later on we got the diesel. In fact, several biodiesel companies simply collect, use the restaurant cooking oil and convert it into biodiesel, which are more existing, the more recent promise of biodiesel generating algae. There are algae which can be having more lipid, can also get the diesel out of it. It will come back to that. And we can modify, in the fourth generation, we can modify some bacteria strain to produce it. Scientists are even genetically engineering this to create a biodiesel lipid, which minimal or no feedstock required, just a sunlight and a carbon dioxide, like a photosynthesis. So with this sunlight and carbon dioxide, we can produce a lipid in a bacteria and extract it and can produce the biodiesel. This is a total cycle. When you go to the oil crops, so maybe sunflower, non-edible oils like a fungemia. In Karnataka, particularly, Assam district, there is a biofuel park. In Madanur, the place called Madanur, you can visit also. So those places, actually, the farmers, now they collect the seeds of fungemia and any other non-edible oils. They can use it and extract the seeds, produce vegetable oil, refine it, and go the trans-esterification. Trans-esterification is the process where actually converting oil into a biodiesel and the glycerol is a byproduct. The glycerol can be widely used in the food industries, cosmetic industries. And then the diesel will be purified, then taken into vehicle and done with the vehicle. There are now vehicles which also run by biodiesel. There's a lot of demand in the railway department. They take this biodiesel to run their vehicles actually for the transportation. So this is a total process involved. In the trans-esterification is nothing but they use the ethanol or any alcohol can be used or methanol can be blend. So then under the sodium hydroxide as a catalytic agent. So they'll mix it and allow for 2 days or 3 days, then finally settle. And we get, after reaction, mixing and reaction, we'll get the biodiesel. This is how the biodiesel produced from the oil. The small unit now available. So small entrepreneurs can take this issue and job opportunity will be created by producing the biodiesel. It is also renewable. There is a very advantage of this. Sufficient plant on the algae bacteria can yield abundant biodiesel. And it is recycled. So whatever you are growing and it will be recycled. There's advantage of biofuel. Much of the biodiesel on the market is derived from the cooking vegetable oils or waste vegetable cooking oils. The domestic production reduces dependency on the fossil fuels and boost rural farming economy. This is very, very important now. The most of them you are depending on the foreign for the fossil fuels. So when you less dependency, then you have a more independency and more economically will be strong. And it also reduce the tailpipe emission compared to the petroleum-based diesel, which is cleaner burning and contains no sulfur and eliminating sulfur dioxide emission. This is very, very important. You know, all of you heard about acid rain. So when the sulfur dioxide goes to that, but during the rainy season, the water mixed with the sulfur dioxide and you will get H2SO4. It's acid rain. So it can also affect on that. So this is how we can reduce. There's a major biodiesel what you are today using. It is containing a lot of sulfur. So that is not there in the biodiesel. So probably people thought that is expensive. No, not much expensive. Now they already it crossed the limit. Now the fossil diesel is expensive compared to the biodiesel. So no, it is not a much expensive. Now it costs you around 60 rupees. The biodiesel is susceptible to cold weather and can gel when the temperature drops fossil fuels. So we can use some other modification or we can fix something to heat up and get just like a gas vehicle we are running. While several diesel vehicle engines can run on that with a minimal or no modification. Biodiesel does not work in the standard gas. Yes, we have to use something engine modification or run this. Or blending is the best concept now. Blending of this biodiesel with the existing diesel engine, it cannot need any modification. The third important one is biogas. I told you and the LPG gas what you are today getting will not be sufficient after 2014. So we have to search for alternatives. The biogas is a renewable energy source. It is a gas produced by anaerobic digestion or fermentation of organic matter. Refers to the mixture of different gases produced by the breakdown of organic matter, and there's a many waste product, either municipal sewage, whatever, compost, food waste, organic manures. So whatever can be converted into the biogas. And there are vehicles which can also be run using the biogas, and biogas can also use widely to generate electricity. So how are you generating electricity, or maybe directly used for cooking, eating, and lighting purpose. How to generate electricity? The biogas which can be used to heat has a heating element and heating energy and heat the water, will get the steam. Steam will be used to rotate the turbine so we can get the electricity. When you look at the cycle, look at that, whatever the sunlight is there, photosynthesis, the corns or the fodder which fixes the photosynthesis during the photosynthesis, and carbon is trapped and they can be used as a fodder, and then it can be waste organic manure, can get it, and then also the organic waste when you do the soil test and other things on the manure, it can be used in anaerobic digestion, and whatever vegetable waste which left out in this fodder can be used as an anaerobic digestion. So after that anaerobic digestion, you get a biogas, can generate electricity, and you can generate heat if you want, and then you can, whatever the leftover water or the carbon dioxide will be exchanged. The carbon dioxide and water will go back to the atmosphere, and again the oxygen will come back to us. So this is how even the leftover after the anaerobic digestion, the methane gas is used here. The leftover is a very good fertilizer. It is free from pathogen, free from weed seed, and is a good manure actually. You can take again to add to the crop and get a good crop on that. This is in a biogas processing technology, actually slurry, but same thing. It is a small type of unit which can be included in this. Okay, this is a small unit and having a lot of impact on that, and also used very widely. This is a biogas plant, which is also this is a small plant. We can look at that. Can be used for cooking or heating light systems, electricity, and also the biofuel is a very good manure. And this is a kitchen waste, animal manure, and agriculture and industrial waste. The biogas is eco-friendly, and biogas generation reduces soil and water pollution. You know that a lot of waste which are there in the system, now we can be reduce the waste and others. The biogas generation produce organic good fertilizer. It is a free from all pathogen, I told you, weeds and other things. It is a simple and low cost technology that encourages the circular economy. That's the reason why people are having a lot of things. All of you know that the waste is going on, and when it's your waste or a municipal waste or in a, whatever the waste which are there, if you expose to the environment, it will produce only hydrogen sulfide. You get a bad smell on that. If you do properly anaerobic digestion with a proper condition and the microbiome, you get a lot of methane. Investment when on a small scale, small biodigester can be used right to and utilizing kitchen waste and animal manure. The healthy cooking alternative for developing areas. It is a very good actually for cooking purpose. It is a developing country like India. We can use this biogas plant. This was there earlier. There's a very big scheme before you are getting this LPG gas. So now because of the LPG gas, nobody is using it in the rural side. Actually, we had a earlier we had a lot of biogas plant in rural area. The few technological advancement, unfortunate disadvantage of biogas body is that system used in the production of biogas is not efficient. There's a new technology yet to simplify the process and make it abundant and low cost. So what happened? So in this case, there are technology now available. So we can use as a community-based production of ethanol. We can overcome this one. Contains impurities. We can also have a lot of purifying agent available. We can use the purifying. We can pass it through the water or pass it through the acid or filters. We can purify. It is not a much difficult. Again, effect of temperature and biogas production. We can create an environment where we can around 37 degrees required. So we can create by using your own system, either solar or wind-based system. Less suitable for dense bitumen. Maybe I think less. You cannot use it in the biogas plant in the city because need a lot of area on the plenty of food waste and manure. But you can have community basis. So in apartment or maybe in a community basis, we can start that. Just imagine without LPG gas in the morning, how to go get your food ready. So this next future, you have to think in that line. Whatever the waste you have created in your own apartment or in a complex or a one center, or you can say any village or maybe in a area where it can confine some area, make a group of that, make a community gas production. That is really the most suitable. That is the future energy to replace LPG. The next fourth one is the biofuel. This is very recently come, and people now is a very promising one. It is because of it is a lot of advantage compared to the biodiesels and other bioethanol that can be potentially directly used by the bacteria, and also isobutanol bacteria like Clostridium can be directly used standard gasoline engine with no modification. It has a lot of similarity to the existing fossil fuels. It is also renewable. It's very important for you. I'll let you know that it is renewable where the fossil fuels are non-renewable. You should understand very clearly. Biobutanol is produced from algae or the bacteria. Can be used directly in gasoline engines with no modification. Can use existing pipeline and supply chain infrastructure for distribution. It has high octane level. There's so little of any loss in the fuel mileage because of the high octane level. No corrosive to engine or the pipeline. The domestic production reduces dependency on foreign sources. So come to there are disadvantages. Feedstock are required for production. So no problem. India is a very diverse climatic condition. India is sufficient to produce any quantity of feedstock required for that. Although non-food feedstocks, genetically modified bacterial strains may resolve this issue in future. The production cost is very highly, but it's not a problem when your petrol is increasing. Just imagine, you look at that when you another 2030, The petrol is 200 rupees per liter. No one will afford to purchase middle family in India. The other way, it is by that time it will be around 60 to 70 or 80 rupees per liter. It is cheaper when compared to the petrol. So anyway, the research are going on for young and other things. People, I think, interested in this line. These are the main area we can also concentrate and do the research on this area. Depending on the origin and availability, biofuels, again, production technology of biofuels, they are generally classified as the first, second, and third generation. The first generation biofuel usually also called as basic biofuel, and second and third generation called as advanced biofuels. So in the first generation, most edible and non-edible. In case of edible, they're using cassava, that is sugar cane or molasses or sweet sugar beet, even rice. Even recently heard about it. Maybe 7 months back, the UN government of India approved the spoiled rice or the grain which are spoiled by the insect or the pathogen, so it can be used to produce ethanol, which are spoiled in the storage because of the lack of a storage facility. Like any waste vegetables, other things can also be used. The next one is a non-edible. There's a lot of oil available, like a pangemia, tohu, and also so many, subabu. So a lot of trees available, tree crops can also available. You can use that for, it's called first generation non-edible oils, inedible or maybe non-edible. The second generation mainly from cellulosic ethanol. 80% of the biomass which are produced on the world is burning, are not properly utilized. You can use this biomass to produce ethanol, that is called cellulosic ethanol. All of you know that in Delhi, there's a lot of fog. The reason for more fog is, one of the reasons is that because of there's a lot of particulates in the atmosphere. The reason is mainly because of the burning of paddy straw in Punjab and Haryana. People can collect it and make a system to collect all those things and convert it into cellulosic ethanol. And like that, you can collect it in the storage. All of you people know that forest fire. You can collect those grass which are grown between the forest trees and convert it into cellulosic ethanol. These are the scope for that to produce. Next one is the third generation algae. Algae is a widely cultivated. You know that it's a very highly productive compared to even any crops. 30% more efficient than any other crops which are produced. We can produce the algal biofuel very easily. There are horizontal and the vertical way of that. Let us come to the first generation. So it is mainly first generation, mainly you are using the food crops like sugarcane, sugar beet, maize, sorghum, wheat, and even rice. We can add the rice also. All the other forms of the biomass based on may even be preferable. You can use other crops also, cassava and other crops, or even potato can use. Problem is the biomass energy derived from first generation source highly linked to provide a long-term substitute for the fossil fuel. And this is the only way we can substitute with the fossil fuel. Again, concerned about the biomass previously destined for human consumption being diverted to fossil. Now it is not a problem. I told very clearly, actual yield is only 35 tons. But actually, productivity capacity, we can grow up to 60 tons. So only 50% you are using, another 50% is not used efficiently. So when you get a good price on something, you can get more, you can grow more. That is not the problem. Concerned about the inefficiency of the first generation. So because our technology innovation is not a much problem, we can overlook in this. Concerned with lower energy content of first generation. Cost also cheaper and eco-friendly and renewable. When you consider all those cost and other things, it will be most important. This is the first generation fuels, just a biodiesel, biogas, whatever. The second generation derived from the feedstock not traditionally used for human consumption. So nothing will be used. Let's take a non-edible compound. This is another way the key player in a broader energy mix. The benefit of using this second fuel is manifold. If you come to environmental side, more environment friendly. If you use this second generation, it is a produce less greenhouse gases. And economics, the produce at a more competitive price because the straw material is worked out, the input cost will be very less compared to the first generation. Make use of vast majority of feedstock, avoid the kind of waste inherent and productions of first generation. So let us come to that in the second generation fuel, how to produce ethanol from biomass. So there's a lot of new technology available. The feedstock which come from grasses, any food and lawn waste can be used, make a powder. Using the enzyme, break down the plant cellulose fiber into glucose and the other sugars. There are three different ways of thermal, biological, or the chemical way of hydrolysis we can do it. Then once you have feedstock, feed to the fermenter, where using the microorganisms like yeast and other bacterial consortium, convert into that and get the sugars, then sugar to ethanol. So then there is a first process, then second fermentation is a pre-fermenting where we can do the hydrolysis and separate the lignin. Lignin will be collected here and you can use for lignin as a burning entity. Then can have fermentation, produce ethanol. Then after ethanol is produced in the fermentation unit, go for distillation, where distilled with high water temperature. So then ethanol will be collected. So the fuel grade ethanol is separated from the solution and distillation and transferred to the blending. Here the H2O is recycled from distillation and separated planting is burned for energy to run the facility. So this can be used to burn and get the electricity. So like you can recycle entire concept, just a grad, this will be converted into a energy or fuel. Next is the third generation biofuel, algal biofuel, also called as oil ghee. Oil ghee is called algal fuel. The third generation biofuel is a fuel from the algae. Algae are low input, high yield feedstock to produce biofuels. It produces 30 times more energy per acre than the land crops. There is an advantage of algae. Sometimes I told you already, second and third generation called as advanced fuels. This is a biodiesel production, is a vertical algal growth. So where the wasteland is available, you can grow the algae in the open tank and collect, harvest, pre-treatment with the compound, chemical compound, extract, and finally we'll get the liquids. Liquid again transfer esterification, we'll get the glycerin and also get the biodiesel, so that go to directly. So during the lipid extraction, we can also get some nutraceuticals, value-added products, vitamins, nutraceuticals, and hormones, whatever. You can get it very, very easy way of getting it. So this is how the biodiesel production process from the algal. This is another way of that. This is actually what you have seen earlier is the horizontal, and this is called vertical. You can produce a lot of algal growth with a short time, high oil price, advanced biotech. Over the past decade, they fueled the algae and biofuel. So same procedure, the algae grow with the sunlight, water and carbon dioxide with a mixing of all these things. With the help of sunlight, it take and grow. So this is the extraction of oil. Around you can get 70% of the oil from the plant, algal oils, we get it. Then solvent used to separate the sugar from oil, solvent then evaporate, and oil is ready and be used for directly in the diesel engine or refined further into biofuels. The fourth generation biofuels make use of novel synthetic biology tools with just emerging the basic research we can have. In the fourth generation biofuel, look at the carbon dioxide. So where we can engineer the crop now. It's a very important. the biotechnologies are playing an important role in the fourth generation biofuel, where they have to genetically modify the crops for high biomass yield, low lignin content, and enhanced carbon storage capacity. That is, does it possible? Maybe students have a think on that, but it is a possible. So we already have a downlining, but with the help of gene editing, we can control the lignin biosynthesis pathway. It is done in one of the crop, lucerne. In a lucerne crop, they have done it. Lucerne crop. They have done it in the lucerne crop. It is possible in all other crops also. We can reduce the lignin content. So it is not the technology is already standardized. We can use any crop reducer. Lignin is the main drawback in cellulosic ethanol production. So we can go pyrolysis, gasification, and digestion. So then a carbon dioxide captured, we can deplete oils, fields, unable water again. Then fuel upgradation, gas cleaning, liquefaction, ultra clean carbon, and finally we get a biohydrogen, biomethane. So these are called synthetic biofuels. So this is how the algal growth we can get a view. This is another way. This is a plant they are engineered, and we can also do the same thing for the algae. It is a very easy in algae. And fourth generation biofuel, where the biotechnologists and the chemists or scientists have a lot of role to produce the bioethanol. It's a design of photosynthetic microorganisms to produce photobiological solar fuels. Earlier, we never thought you have a silica-based solar cell, silica cells we have. Silica cell, it is not manufactured in India, again it is imported from other countries. So when you think of photobiological solar fuels, so this is a best way to produce solar light, harvest the solar light. The combining photovoltaics and microbial fuels in production are by synthetic cell factories to synthesize organisms, specifically tailored for the production of desired high-value chemicals. The one way you can convert. There are lots of ways to create things for that. The fourth generation technology combines genetically optimized feedstock, which are designed to capture large amount of carbon with genomically synthesized microbes, which are made efficiently to make the fuel. Key to this process in the capture sequestration carbon dioxide. The process that render fourth generation fuels are also called as very important one is carbon negative source fuel. The fourth generation biofuels are called carbon negative source of fuels. That is a requirement for us to control the greenhouse gas emission or the global warming. This is a total technology how we can produce the biohydrogen. Biohydrogen is one of the source of energy now. So we can even use in the vehicle, and run the vehicle. There are three ways we have: biological, thermochemical, and electrochemical. Biological, again, the modified microorganisms by the fermentation or photosynthesis. These are the way we can also produce. Thermochemical, we can use gasification, partial oxidation, stream of reforming, cracking, and pyrolysis. So this is the photoelectrolysis and electrolysis. These are the way in which we can produce the hydrogen. So what is fuel cells? Fuel cells is the device that converts the chemical energy from a fuel into electricity through chemical reaction with oxygen and another oxidizing agent. So that is the definition for fuel cell. This is the hydrogen fuel cell form. It is a very famous one. You know, Indian origin scientists in America had developed a fuel cell pack where it can generate electricity. And particularly, he developed a unit similar to a refrigerator. If you keep it in your home with a refrigerator, with this fuel cell, you need not depend on electricity for any other source. The entire fuel cell provides electricity to the entire house requirement. So it is patented. Probably we also get shortly to India. So there's a lot of scope in that. But the source of energy and recycling also varies with the stuff. The overview of this biofuel production. The first generation, I told you, starch, sugar, oil crops. This is called first generation, where maize, sugar cane is missing here, but potato, And even what are the crops which are rich in sugar? Even oil crops can also be used in the first generation. In the second generation, where you can use the straw, nut cells, manure, and even crude glycerin can be reused here. Municipal waste and industrial waste. You know, a lot of waste are available in Bangalore. How to convert into fuel? It is a very important and the task force. You can do. Third generation where actually algal biomass. That is important. The fourth generation where specially designed microorganisms, genetically engineer algae and the plants. So those two can be modified and can generate the carbon negative is a very important one. So the day it will come, we'll have seen fourth generation fuels also. If we come to the biofuel, so what you thought of most of them, you're now searching for government job. You have less opportunity. So why can't go for a part-time job? You can become entrepreneurs by doing a part-time job also. Entrepreneurs means you are independent. You can do part of your time, can you spend how to produce that. This is only for youth searching for job or maybe a teacher working on contract or low salary basis. They can start your own entrepreneurship in the place that like that. What is there in that? Three top reasons why entrepreneurs should go for biofuel production. Because it's a job opportunity for the people who are looking for job and other things. The first one, the world wants to reduce the waste and recycle more. You know, a lot of waste are available. We can have to think on that, can collect it and recycle it. That's one of the way, reduce the waste and recycle the waste is a very, very important for us. There's a growing preference for cleaner and eco-friendly fuels. Probably you know that burning of petrol and diesel emit the carbon dioxide. The carbon dioxide level reaches 600 ppm. So it is increase the temperature. Then we cannot live in the atmosphere. So there's and also a lot of problem with that other things like ice will melt and the water levels in the sea will be increasing and some of the cities will be submerged in this water. So these are a lot of impact were there in that. And this is the reason why the entire world is looking for that. So how to control this emission or reduce the emission. That's why people should go for the eco-friendly fuels. More countries want to reduce their dependency on crude oil products. Because the economy, important is economy. You are importing. You are spending around 12,000 crores money. Just imagine, you cannot do so much of 12,000 crores of money you are importing for oil. There's a foreign exchange, you lost 12,000 crores of work every year. So you can be revealed. There's a most country need to have that diversification. The foreign investor, for example, in Tanzania, a jatropha grower started a company and collect the seed and produce ethanol and become a good entrepreneur now. So like that, there are people, you can use the wage or whatever they invest. So you can get a lot of money for a period of 6 years. You'll get back your money. So this is the way entrepreneurs are done. There are successful entrepreneurs also in that. The role of biotechnology, encourage entrepreneurs. So I think most of them are biotechnology students. So end-to-end solution for biofuel. There's a lot of opportunity. And creation of awareness of the product. Rising of seedlings and planting activities, training program, introduction of new and promising biofuels. So there's a lot of things to do and to encourage the people on that. Advantage of biomass energy. This is another way in which, forest of them, you're getting a forest fire. During forest fire, there's a valuable timbers will be burned in during the forest. It is not only for India, it is in developed countries. The reason for forest fire is because of the biomass or the weeds which are grown in between the plants or in between trees. Even in India also we can see. So what happens? During the summer it dries, it catches the fire. So what you have to do now, you have to collect either during a wet condition or maybe before it dries, collect those weeds. And we can convert as things. We can generate electricity out of it. 30 to 40% of energy requirement, electricity requirement can be generated in situ in the same place. In every taluka, the district, we can generate. This is an advantage in new area People can have invest in this area. So for example, you can have wind energy or sun or maybe idle energy. Our biomass energy required less money compared to this wind energy. There were 6 crore, for example, to produce megawatt of electricity. The biomass energy before around 6 crores is required for gasification and or maybe other way 5 crores in Rankine cycles. So this is some of the way you can also have a biomass energy. Biomass means generating clean energy, generating income, and there's a lot of flexibility and scalability is possible with this energy. Uplifting of villages by decentralized distribution and relief. When the energy requirement, electricity requirement is decreased in a village or in a taluk, so you get more electricity available for urban area and with a cheaper rate. Because it is a costly because of that. Then good quality electricity available to the villages, sufficient local generation and more job opportunity for this. Because otherwise the people are migrating from rural to urban area. So it creates a lot of problems. Pollution or maybe traffic, whatever. So that can be controlled by the setting up the decentralized distribution of the place of that. So energy plants can be located in any rural area and can do things like that within the radius of 25 to 40 kilometers can produce 1 megawatt to 5 megawatt. So this is whatever the waste which are available can be recycled as a biomass. So come to the biofuel policy. So the government of India has started a national policy on biofuels 2018. So it is now also good. I think I will come back to the next picture. I will explain you. This is a national policy on biofuel 2018. The important features of this, an indicative target of 20% blending ethanol in petrol and 5% blending of biodiesel in diesel is proposed. If you do this much, it's really a big contribution to economy-wise and also environmental benefit wise. It's a major contribution from this biofuel. So with the trust of advanced biofuel, the policy indicates the viability gap funding scheme for 2G ethanol biorefineries of 5,000 crores in 6 years. The government has invested so much of money. In addition to that, they also give a tax incentive, high purchase price compared to the first G biofuel. First G is first generation biofuel. And 2G, the second generation, they're giving a lot of importance. So always use the non-edible product for that. Then basic biofuels where the first generation bioethanol and biodiesel, and the second generation is also called as advanced biofuels. And algae-based is called as third generation biofuels. Increased scope of raw material for ethanol procuring by an encouraging intermediary. Apart from B-grade molasses, the molasses is a byproduct of sugar industry. After getting your sugar, whatever the waste goes is called molasses. Apart from molasses, they can also use the sugarcane juice, other sugar content materials, and damaged as well as the surplus food grain. That's what they included. The rice can also, broken rice or damaged food material can also be used again. And now they start develop the national biomass repository by conducting a price of biomass across the country. Biodiesel production to encourage from a non-edible oils using a used cooking oil or short gestation crops, development of supply chain mechanism. The trust and research development and demonstration in the field of biofuel feedback, production, advanced conversion technology from identified period. Setting up of a National Biofuel Coordination Committee, NBCC, under the Ministry of Petroleum and Natural Gas and working group of biofuel. So these are the things of biofuel policy, national policy in 2018 has been made. And now because of the COVID, not able to do anything, maybe further qualification will be done in the future. Even the additionally World Biofuel Day will be celebrated on 28th of August every year. Probably I think there is a World Biofuel Day. This August 28th, so they are celebrating. It is also celebrated jointly. The recently they started. So then to stem fall in the farmers' income, government approved, and Modi has said ethanol will help cane farmers to find a permanent solution. He said the use of biofuel will improve farmers' income, create new employment opportunity, and save foreign exchange for the country. This is the same thing what you have Bioethanol can also be done by using the waste biomass, likely rich in crop residues like the sugarcane bagasse, corn, maize, stover, rice straw, wheat straw, and the rice bran, sugar-rich product like a sweet sorghum, byproducts, sugarcane waste, and other things. Even waste paper, probably all of you know that there's a lot of waste paper. The writing paper which can be used, hydrolysis and converted into sugar, then into the ethanol. So this is called lignocellulose feedback for sugars and ethanol production. Coffee residue waste can be used. Municipal solid waste, biosolids, sludges, livestock manure, food waste, and wood waste. So a lot of availability. So one way, when you use this waste, we recycle the waste material. And you are cleaning the material, the area also. The cleaning way also, there is another way of adding contribution to the eco-friendly environment. Example of conversion factor, 27 kg of soybean yield 5 kg of soybean oil, making 5.7 liter of the biodiesel. Similarly, if you go to the 25 kg of corn yield 9.5 liters of the ethanol. The 1 ton of, around 1 ton of corn stover yield 300 to 340 liters of ethanol. And also similarly, grass can yield 285 to 380 water. If you come to the overall economics, around 380 million liters for ethanol production facility could create 2,250 local jobs for single family. Even 1% blending, if you do for biodiesel blending, they are given for 5%. If you do 1%, required around 1.1 billion liters of biodiesel. So that will provide around 56,000 million rupees gross income added to the farmers. So if you do ethanol production, so required around 8.9 billion industrial output and 3.8 is a billion. It create around 76,000 permanent jobs. So this is a biofuel is creating a lot of jobs for people. So environmental benefit, okay, apart from employment generation, economic benefit, we also have an environment. That is a very important point. The biofuel blend with the conventional fuels reduce air pollutant emission such as sulfur, particulate, carbon monoxide, and hydrocarbons. Ethanol and biodiesel are also less of a jar if they spill or leak since they are rapidly biodegradable in water. If, for example, petrol, diesel spill out, it kills a lot of animals, worms, or insects which are living in the water or in the soil. But it's not the case of ethanol and biodiesel. Substituting biofuels for 1 gallon of gasoline of diesel save up to 20 pounds of carbon dioxide emission. Just imagine to the atmosphere since they are made from carbon recycled by plants. Instead of carbon dug out of the ground in the form of fossil. In a fossil way, whatever you have burned it, petrol, diesel, LPG, whatever you have burning, so it has a carbon dioxide to the atmosphere. And the carbon dioxide level is increasing. Every year, 2 degrees centigrade of temperature is increasing. The growing the perennial energy crop in place of surplus annual crop can reduce soil erosion, compaction. A permanent deep root system develop the enrich the soil and reduce the carbon dioxide level in the atmosphere. So there's a reason why I request all of you, at least during your birthday, every birthday at least we can grow 1 tree. So if you are now, I think you are young now, maybe 15, 20 years, another 20 years, at least 20 trees in your life, save a lot of help, particularly a lot of benefit. So if you don't have a land, I can go to some common place. Plant 1 tree in your birthday. Take care of till 1 year. It gives a lot of benefit to us. So plant a tree during your birthday. I'm doing actually since I was studying 8th standard, since 8th standard. Now I've grown more than 200 plants or maybe it is more than that. So it is required actually all of you. I think we don't have space, at least plant 1 tree in your birth, in any place, common place, in a temple, church, wherever you are. Go and ask them, request them. This is the place where I'll grow the tree. You can grow any tree as you like it. Any fruit crops, jackfruit or something. So that will provide revenue also. So I request all of you, I hope you can do for that way. You can grow the tree, which a lot of environmental benefit, all of that. Conclusion, finally, the biofuel in India is a strategic importance for ensuring India's energy balance and security. Energy security is the main lacuna in India. If you have a good security, we have food security, we have fiber security. Now you need to have fuel security. So if you have fuel security, India economically grow well and will stand alone. No need to depend on any other country. I told you very clearly, we are spending like 12,000 crores is the foreign exchange money if you go with the biofuel. So even not only making the biofuel, make the Swachh Bharat Abhiyan, whatever the government has created for. So that will also be doubling the farmers' income, import reduction, employment generation, waste to wealth creation. Simultaneously creating a win-to-win model for people, profit and the planet. Not only you are getting profit, and also you can save the planet. So love your environment, only way to save it. And thank you so much. And thank you so much for the host institute for providing the opportunity to deliver this lecture. Thank you so much.
Thank you, sir, for the very informative talk. Questions can be put to sir. There is one question in the chat box, sir. I'd like to read it to you.
Yeah, please.
So this is from Anirudh. He thanks you first for the wonderful presentation. And he goes on to say that his opinion about biofuel is that it can't be a global success because crops can't be grown in every part of the world because of various climatic conditions. And some places like arid area, there is limited water resources, since water is the most important component of biofuel. Plus, as you also said, sir, that byproducts such as hydrogen and carbon dioxide could cause global warming. Why? So what do you think can biofuel be a much more sustainable fuel for transport than electric, solar, nuclear, or hydrogen fuel cell? Can it be the fuel of the future?
Yeah. So we'll come to the first question. In the area where we not have much water, we can grow some of the crops which have less sustainability. So there are crops which grow in a less water condition. Like so, there are crops available, forestry with seed, actually oil seed crops. The first option. Second, still is the worst come. We cannot grow anything. Go to solar, harvesting solar energy. That is also renewable energy. We can go to a solar panel and create electricity. Or still we have a lot of extreme condition, hill area cannot grow, and we can go for windmill. So we can have a wind also. So where we cannot grow, we can have a lot of, these are all are renewable only. So that's the reason why we can go to solar based energy or wind based energy system. That is not a problem. If you really have a very less water, we can go for some crop. Actually, go plant a tree. You can grow very well. You take care of only thing small time you have to require for that. I'll give an example. When I was in Israel, they've shown one hill. Actually, they've shown another photo also. Put it there, another photo. That photo was 20-year-old. But now we've seen there's a thick forest. Nothing is there on that. Not even a grass grown on that forest. But they have taken a drip irrigation and grow the entire hill. Now it is a full of covered forest. It is a possible now. It is not a much difficult, not so difficult nowadays because of the advanced technology we have. We can do things. The second one, nuclear. You know the impact of the nuclear. We can also supplement, and solar or whatever wind energy is also be supplement for that. But you cannot say 100% cannot depend on that. That's why the biofuel is one of the major source. It is also supplemented with solar or wind energy and also nuclear. Even though I think the Chernobyl in Russia, there is a disaster happen because of the nuclear. Not only in Russia, even in Japan, you've seen that nuclear approaches will create a lot of nuisance and create a lot of problems also. It affects not only human beings and animals and the environment, also affected with that. So compared to that, I think it is the way we can use it. Thank you, Anirudh.
Okay. If there's anybody else who would like to ask questions, please ask, sir.
Okay.
Excuse me.
Yeah.
Myself, Dr. Pankaj Kumar Singh from IIT BHU. Regarding this topic, the biofuels, we have given a very nice lecture. I am having a small query. As the biofuel plant is taking a large amount of land, that may be a problem that
Okay, we'll come back to that. No, you know the agroforestry now, people are catching
that
up.
is first problem.
Yeah. So, you know, lot of space is available now. People not efficiently use their land at all. When you travel from any place, to one place, now we have stopped to travel because of the corona. Earlier, I used to travel from Delhi and other things. I always look, take a window side, and I used to look for the place where actually there's a lot of areas available. No problem. I think so. India has a lot of problem. Look at the lot of area vacant. You can go with the forest, agroforestry system, so where you can grow crop in the middle whenever off season. So then we'll get in the trees, we'll get oils and non-edible oils like fungi and whatever crop which are grown in your area. You get crop oil.
Second is that, yes.
The second one is the productivity. You take average of crop, any crop field of Indian crops, we have only exploited only not even 50%, less than 50%. You have an opportunity to have for another 60%. I told you this example, sugarcane. 35 ton is the per hectare yield in overall average India production, but the potential is there 60 ton. You have just exploited only 35 ton. There's a lot of scope in that way. Okay. Yeah, Pankaj, any question?
Excuse me. The second one is it is using the crops that is taken as food. It will also create a problem.
No, not at all. I'm telling. I made a lot of survey on that way. Then not at all it affects on the food because farmer, go to village. Village now become a senior citizen home. Farming is not economically profitable because the less price they are getting. So there's a lot of lands which vacant, not able to cultivable. Even the irrigation is there, but not able to cultivate. That's the main reason. It never affect on the food security. Same thing happened. I'll tell you, maybe 10 years back, when America start converting maize, there's a maize with weedicide resistant maize, herbicide resistant maize. They have converted into ethanol. Now India, they have the more than 40% contribution from America only. It is not there in 1996, 1986. But today, America stand number 1 in the world because they're converting the entire maize or corn into ethanol and the animal feed. Nothing happened. Even entire world looked at and commented on that, but they are not bothered. They tell, no, it's not affect that. But today it's true. No one is suffering from food. It never affect. Actually, the excess production, whatever you can do, we can do. No, it will never affect the food. That's a wrong concept. People are telling that. That is not correct. Thank you.
So there is one more question on the chat. I'd like to read it. This is from Shubham Mondal. And he thanks you for your super presentation. His question is, what happens if a car is run on biofuel instead of regular diesel in a car which should have regular diesel engine?
No, at present, I think we are thinking of blending, but there's a modification is required. So you can run only on these, just like a biodiesel. What you have a fossil diesel, similarly, you can also run with the biodiesel, but need some modification in the engine. Need some modification in the engine.
Thank you, sir.
Thank you.
I hope there aren't any more questions. And thank you, sir, for the excellent speech. And we may now head for lunch. And let's all be back by 2.
Excuse me, sir.
Yeah.
Sir, there is no any feedback from morning.
Answered your question on the chat box that there is going to be a feedback. I mean, a comprehensive feedback at the end of 4 sessions today in the evening, sir. Covering all aspects of the 4 sessions today, you will have one feedback in the evening, sir. I have already put the answer in the chat box, sir.
Okay, thank you, madam.
Welcome, sir.
Okay, thank you. Thank you all. Thank you, Prabhakar, madam. And also thanks to my beloved teacher, Dr. Bhagara sir, because of my guide who has tuned me to become a teacher and a scientist. Thank you, sir.
Thank you so much, sir. Thank you for your time and for sharing your information, your knowledge, and your experiences with us, sir. Thank you so much.
Yes, sir.